Welcome back everyone. I hope you all had enjoyed your lunch break. Thank you for joining us for the next session of the SNCI Neuroscience Symposium 2025. We will continue with the series of engaging and the insightful sessions. So moving forward, it's my honor to invite Dr. Anurag Varsne sir, who is currently leading Patanjali Research Institute as a Vice President, Drug Discovery and Development. Dr. Varsne is a seasonal drug discovery and development professional with 24 years of the research and the managerial experience. He has done a PhD from the National Center for the Biological Sciences, TIFR, Bangalore, postdoc from the Yale University School of Medicine's New Haven, USA, and MBA from the IP University Institute of Management, New Delhi. Before joining Patanjali, Dr. Varsne had worked in different capacities at the Marine Biological Laboratories, at Ranbaxy Research Labs, and at Daiichi Sankyo Pharmaceuticals in multilingual multicultural environments. He has driven more than 50 diverse research themes with specialized risk mitigation and effective resolution capabilities. On personal traits, he is seen as a multi-compartment thinker and the natural accelerator of the science and its business. Welcome, sir.
Very nice. So I can see the food was good and there is a good amount of carbon dioxide in the body. So good afternoon is also muted. So all the students say good afternoon.
Good afternoon.
Yeah, now we are ready. So this is good. So my name is Anurag, and after having such a long introduction, it becomes even more challenging to meet it, but I will try that. So I come from Haridwar, one more holy city where the Ganga starts and all the way comes over here, and I come to one more holy city of Varanasi, and it feels good connect. I came last night and I got welcomed by very good rain, and the guest house was fantastic, food is even better. After a long time I had parwal aloo cooked in a eastern style, which was very good too. So as you can see by my dimension, I am foodie, I love food, and good food drives me far away, so I felt very happy. So thank you very much all organizers for setting this up and doing this science-driven conference, which is very, very critical. So I have been part of the neurochemistry since my PhD from NCBS, and we have been working on lot of different components. By my PhD, I'm an electrophysiologist, have done a lot of neurophysiology, and chemistry have been part of it. And during my graduate days, maybe 20-25 years before, we used to call chemical locha in the brain, which actually is the neurochemistry. But today I'm not going to talk more on those. Today I'm going to talk more about Ayurveda, and in morning we have two fantastic lectures. We had very good understanding about how yoga could help. Dr. Rao really talked so well about it. Some of them were very eye-opening. I learned two new words from him today. I learn every time whenever I see him. This time he gave me a new word called hypernutrition. When I read this word, it reminded of me. So I actually learned that much from him. And second, he gave a very fancy tongue-twisting name, which is being used by people outside India, pranayam ke liye. Bahut acha wo kahiye fancy word. I wrote it down, but it was good as well. So before I start my presentation, I would like to, of course, thank the organizers. It was really nice. I am very thankful to Professor M.P. Thakur sir, the President of the Society, Professor Rajesh Kumarji, the Dean of R&D at BHU, and the great man himself, Dr. Rajavan Raoji, the Director CCRN, who has been behind the basic science for yoga and naturopathy across India. The amount of efforts he put and the amount of energy he invests and the amount of travel he does, I'm sure he's done a lot of frequent flying miles. I don't know how he's going to use it, but he does this marvelous. Because there are several councils in Ministry of Ayush, and whenever I see this, because I interact with many people, whatever comes from CCRN is actually marvelous. So what I'll request all of you, if you still have that CO2 less in your blood, let's clap hard for him. So thank you very much. And then Dr. Vinod Tiwari ji, the convener, and Dr. Rajneesh ji, the co-convener, who have been the young folks who have been behind this and doing it so well. So now with all those starting phase done, let me move toward the science I'm going to talk about. So by training, I am a biomedical researcher, and I talk about research. So for me, using a small molecule, it's a testing article. Ayurveda, the drugs become a test article. So my life before coming to Patanjali was into pharma industry. For like 12 years I was working with one Japanese pharma, Daiichi Sankyo, for Indian pharma MNC at vaccine. And there we were doing drug discovery for modern synthetic drugs. Now at Patanjali things have changed a bit, but not much, because the test articles earlier which were coming from medicinal chemists, now they come from nature, but they come from herbal chemists. And talking of herbal chemists, I'm sure there are many sitting here, and we have a couple whom I know very well. There's one girl there sitting in the audience who have been the herbal chemist at Patanjali, and now she's refining her knowledge and going towards PhD. Ms. Katak, who has been doing very good too. And when I said कि I would be coming, she wrote me a message कि सर अगर आप आ रहे हो एक बार मुझसे जरूर मिलना, तब से मिल ही रहे हैं तो ये बहुत ही अच्छा है. अब हम बात करेंगे Advancing Ayurveda Neuroscience Notes from Invertebrate Models of Neurodegeneration. तो बेसिकली जब आप किसी एक दवा के बारे में बात करते हैं, मेडिसिन के बारे में बात करते हैं, तो आपको उसके मोड ऑफ एक्शन के बारे में बताना है. You can do clinical trials. We heard a lot from Dr. Banerjee about how we can see the effect of yoga on clinical conditions. But now I'm going to switch a little bit. I'll go to what effect of Ayurveda and doing evaluations in different animal models. We conduct a lot of research on different animals. Today, I have selectively taken invertebrates, animal models which do not have vertebral columns. And those can also be used for looking for the basic biology behind it. And I come from Patanjali Research Foundation, and in the next few slides, I'm going to introduce the center where I work with, because most of you may not be knowing about it. So I thought I'll take 4 or 5 slides to explain where I work and what I do. So these research institutes are fairly new. This was set up in 2017 and inaugurated by our Honorable Prime Minister Modi ji. These ultra-modern laboratories have been set up to bridge the gap. The theme of this conference today is also talking about the bridge. So this bridging has been planned and implemented and executed at Patanjali to fill the gap between the modern practice and our ancient medicinal knowledge system of yoga and Ayurveda. It is probably one of the only kind literally state of the art. Because we scientists, for us, equipments are toys, and everyone wants toys. We get more toys, more happy we become. So we have this kind of leverage, freedom, and little bit of over freedom too, to use the type of technology that we want to work with. So top of the line equipment, globally trained workforce, energize operations on a daily basis. And like every other institute, we have a mission. A mission for our institute to improve the health and quality of life through research and innovation. And to do that, we have the vision. So the vision is to take all the unstructured Ayurvedic herbal information with the rich history of medicinal plant, go through the funnel of scientific methods combined with old wisdom, modern practice, fantastic scope of delivery, multi-pronged methods, and you have something what we call research Ayurveda. In the end, what we are aiming to get a scientifically validated, evidence-based medicinal system which is accepted by Ayurveda as a system for treatment, which only happens when we give the evidence which becomes acceptable by everyone who has to do that. Now, when we talk about research, it becomes very critical what kind of accreditations you have, what kind of setup you have. And the slide that you see here, slightly busy, but talks about research strength and accreditation in Patanjali. So the laboratory that we work with, it has annual accreditations, which is of course normal to get it done. But what is more critical, that we have biosafety level 3 and level 2 laboratories, which are very clear concepts of maintaining corridors for doing the research for biology as well as for microbiology. This is accredited by DSIR. Close to 300 scientists work there, several PhDs postdoc from global experiences, and the whole team is now actually channeling towards getting Ayurveda into the right direction. We have a full-fledged registered animal house with CPCA, which is driven by Institutional Ethics Committee, and we also have a herbarium, which is accredited by New York Botanical Garden in USA. And we conduct a lot of clinical trials. We have ethics committees for conducting trials, and most of trials we conduct are driven by CTRI registration as well. Now, what is good, that we have a hospital, we have an Ayurvedic medical college just next door. So we have folks who are doing their PGs in Ayurveda, and I think close to now 9 subjects. What the senior is 8. There's one more update, and 250-bed Ayurvedic hospital with MBBS accreditation just next door. And to make it things more complicated, close to 2,200 Ayurvedic doctors are on the roster of Patanjali across India. Even in Varanasi, there are 3 centers, the 3 Patanjali mega store, where you will have an Ayurvedic physician sitting over there. Those people, they actually see the patients on daily basis. The daily footfall is close to 12,000 patients all over India. And whatever is being seen by those Vaidyas goes into a system. And the system is called Patanjali Electronic Medical Records, PEMR. And the total information on that system is close to 1 crore people. 1 crore patients का all data उस system में है जो पूरे भारतवर्ष से इकट्ठा होता है और ये पिछले 15 या 20 साल से होता हुआ है. Now, when we talk about research, then the point comes क्या क्या करते हैं, what you can do. And all the students in the audience, अगर मैं बहुत तेजी से बोल रहा हूँ तो अपना हाथ ऊंचा करना, मैं हल्का बोलना शुरू कर देता हूँ. I speak fast, sometimes I stammer, so if I get stuck, please pardon me. But if I go too fast, ask the questions, right? I want this to be more of interactive because whenever I travel for a conferences, my intention is to talk to the students because all the folks in the front row, they are pre-conditioned. You really cannot change them much. But what is sitting on the backward has the potential to grow in the direction where they can go, right? Front rows are also very important, right? Because they are driving the research in the right direction. And we talked about Dr. Rao, how he is driving, right? So now, when we talk about research, these are the major 5 areas that we work with. We do drug discovery and development. I'm going to talk a lot about it today. We do herbal research, where we compile the data of all the medicinal plants in the whole earth. And we generate a fancy document called World Herbal Encyclopedia, which goes in 109 volumes and close to 12 lakhs medicinal plants have been listed there in all possible language on this earth with their biological values written, defined, their toxicology written, pharmacologically effect written, all large compendium. It took 15 years of the total time by the scientists to get this document done. And we have a very fancy collection of paintings and herbariums. So close to 35,000 medicinal plants have been hand painted on canvas, and those are there at Patanjali Research Foundation. And I really request all of you, please do come and visit us. You are going to like it. And that collection is actually marvelous. This is the largest in the whole earth where we have such a large collection of medicinal plant painting. We also work with ancient documents, and this becomes a treasure trove. We pick up lots of information from there, close to 12 lakhs old document sheets, manuscript written on a piece of paper, piece of wood, piece of leaves, which have been written by our ancestors. Most of the language cannot be really understood even now. Those have been compiled at Patanjali. There is a dedicated team which actually translate them into a Sanskrit, from Sanskrit to English and Hindi, and they are being now compiled in form of the book as well. And of course, yoga is a core competency of Patanjali. There is a team that takes care of the yoga research too. Now, talking about drug discovery and development, that is my forte. So we have basically 5 major departments that work under that division. We have an in vivo biology, which works on the animals. As you understand, we work with almost all animal models of our choice, starting from very small Drosophila to C. elegans to rat, mice, guinea pigs, rabbits. We do research on dogs, and we also do research on monkeys. Monkeys, things we do in China. And all of these, those are the animal models which become useful for showing the proof of principle for the functionality of the drugs that we work with. We have in vitro biology, which works on basic biochemistry, cell biology, molecular biology, and other kind of interactive things that we work with. Herbal chemistry is the team which works on generating the test article, which does collections, extraction, quantification, analysis, and also the report generation for each compound that we want to work with. Microbiology, it's our next team that works on bacteria, virus, and fungus, and also for post-pathogen interactions. And last but not the least, a very critical one, the clinical research. So we conduct clinical research in-house. Close to 2,500 health seekers are there at Patanjali on a daily basis. We collect data from them, and we also conduct trials at multiple places. So we are conducting trials at AIIMS, at 4 AIIMS right now in India. We conduct a couple of trials in Tata Memorial Hospital in Mumbai. The one we will also start here in Varanasi too. And the recent one just started at AFMC, Armed Forces Medical College in Pune. Now, this is one of the critical slides, because when we start working on Ayurveda, the difficulty happens how are we going to start doing the research. So when I joined in 2018, I had a long chat with my boss, Acharya Balakrishna ji, he's my boss, and we had a like 3-hour chat about how are we going to do. And I asked him how many test articles we're talking about. He said there are close to 1,000 medicinal plants which Ayurveda uses, and there are close to 1,000 formulations that Ayurveda has defined. I am not a student of Ayurveda. I was not a student, now I am a student of Ayurveda. I didn't know much about it. So I said I'm going to take it as a test article and see how we can do the research. So one way is that you take a drug and you see which disease it can modify. That might take longer time. And the other way would be that you take a disease and see which drug can fit into it. It's like winning the drugs. So this is what you're seeing right now. This is the disease-driven approach which we followed, and we actually start looking for these therapeutic areas: inflammation, metabolic disorder, cancer, neuroscience, and infectious disease. And what you see here are the various models that we have developed. This number says that these are the number of models we have developed for these diseases. They're a long list. The team has been working very hard to keep that at base. But what I want to talk today is about neuroscience, pain and diabetic neuropathy, learning and memory, stress, and cognitive functions. So today I'm going to focus a little bit more about the work that we have done on neuroscience. Now, this is one slide which is good for the students to follow. And this will also help you if you want to do something different in your academic life or professional life. Now, here comes the concept of reverse pharmacology. How many of you know what is reverse pharmacology? Raise of hands. Reverse pharmacology? Good, very nice. So we have one gentleman. Let's clap for him first, huh? Very nice. Now, when I come from a pharma industry, the biggest challenge pharma industry has is getting the success rate. What does it mean? That it takes close to 10 years and $10 billion to get one drug from medicinal chemist bench all the way to the patients. Right? The clinical trial success rates are very low. The risks are high, and that's the reason most of the businesses are moving away from new drug discovery. It's happening a lot in India too. So how you de-risk the whole process of drug discovery? And that process is reverse pharmacology. How we do that? So the conventional path is that you synthesize a molecule, which is happening in the pharma industry. You test in animals, mice, and then you go to the man. And that becomes the path which takes 10 years and $10 million. Now, other way out, because the bigger risk is lying here. Most of the drug fails in clinical trials. Phase 1, phase 2 will take much longer time, a lot of money. What we have in Ayurveda or in the ancient texts, or in case yoga as well, that we know the drugs work, the process work in man. For the last 2,500 years, in Charak Samhita, we know this drug works for this disease, and we know the safety because it is being used for so many centuries. So the safety is very well known, which is the much bigger concern for pharma industry, and efficacy is just a matter of validation. So why not we validate of the pre-known safe drug? We already know the data in man for that matter, right? At least that is classically described. And from there you go to mice and go to the molecule. Basically find out which part of the drug engage which part of the pathway in a cell or in an intact animal, and find out the mode of action. Then the acceptability will come. You will validate and efficacy will be accepted. That becomes the part, and that becomes the biggest challenge as well. What you see a triangle over here, that's my favorite triangle. Why? Because it's right now looking impossible. If you run your eye on each arm of the triangle, it will not add up. It will go back where you just started. Because these are the 3 arms where we need to work with. These are called golden triangle approach of drug discovery. You take Ayurveda, the traditional wisdom, combined with modern sciences, which is the technological advances. By saying it, I mean like new technology, for example, a better quality CT machine, better quality MRI, even a better thermometer is a modern technology which was not available to our ancient saints several thousand years before. Combined with this, and then also add modern medicine. As we heard from Dr. Banerjee just before the lunch, that having an evidence-based clinical practice have combining all 3, and you would be able to get this golden triangle done. And since I work for Patanjali Research Foundation, very close to my heart, you would see in the heart of the triangle the logo of BRF. Because we believe that we are at the crossroad of the time and the space, and we would be able to hit it out. Now, here comes the science I'm going to talk today. Advancing Ayurveda and Neuroscience, notes from invertebrate models for neurodegeneration. And this, I think all of you already know this. And Dr. Rao in the morning talked about very well that close to 3 billion people worldwide are living with the neurological condition. That means 1 out of 3 individuals on this earth has some kind of neurological problem. And these symptoms could be either from pain, cognitive difficulties, motor impairment, and all of it. And we also heard that as whatever you pour in your brain, what you become. And as Stephen King said, the brain is a muscle that can move the world. However, there is no muscle in the brain, but brain has those power to move the whole earth too. So when we talk about common neurological diseases, there are several of them. 3 of them becomes very, very famous: Alzheimer's, Parkinson's, and multiple sclerosis. Today, I'm going to focus about Parkinson's disease and see what kind of animal models we can set up and see the effect of the drugs that we have been focusing on. Am I getting too fast or is all okay? All good? Very nice. How many of us are sleeping? None? Good, very nice. Because I learned something very good at Patanjali. Is this: the moment you get bored and the moment you get sleepy or you feel that now bahut ho gaya, khana bahut acha tha, all you have to do, take a deep breath and say Om. Right? So how about we practice that Om as well? Is it okay? I'll go slightly longer Om. Okay? Deep breath. See? It works, right? So now we're going to take it there and see the understanding of the brain and nervous system. We heard from Dr. Rao as well that the functionality of the whole brain is driven by neurotransmitters. Today we talk about connections, right? Connectivity. That connectivity that happens at the level of society also happens at the level of brain. Each cell of brain, neuron, connect with another cell through a connection called synapse. And these synapses are basically physically apart. Between them there is a small cleft where some chemical passes through. Those chemicals are neurotransmitters. So we have dopamine, serotonin, glutamate, and acetylcholine. And all of these 4 are the critical ones. There are 2 more. But these are the ones that play a significant role. And deficiency of either of them causes a neurological disease. For example, dopamine deficiency will cause Parkinson. Serotonin deficiency will cause depression because serotonin is a happy hormone. If you're happy about it. Glutamate, epilepsy, acetylcholine, Alzheimer's. So today we're going to talk more about Parkinson's. Now Parkinson's is the second most common human neurological disorder. It affects the 1% population on this earth, which are 65 years and above. And this generally happens because of degeneration of the nerve cells. And we heard in the morning that once the degeneration happens, it's very difficult to come back from there. And this happened in this area, which is SN, substantia nigra, where this degradation happened. And the dopamine deficiency, which is required from one transmitting neuron to a receptor cell, becomes low, and that becomes a molecular hallmark, a molecular path for how this disease gets induced up. Now this is a very critical slide, and that made me realize a lot. This is the data that came very recently from one of the sponsored study of WHO. These are the years we're talking about, 1990 to 2050. These are the countries which have been listed who would have Parkinson's disease at that time. And what is the projection in 2050? You see the top countries who would have more patients of Parkinson's are China, number 1, and number 2, India, followed by US. That means our country is more predisposed to have patients with Parkinson's disease. That would mean it becomes critical that we start thinking about it now. And that is a very sizable number because India is going to have close to 2.8 million patients by 2050 having Parkinson's. And the pathogenesis, how does it happen? At a molecular level, we must understand how the disease goes. So there are mutations in multiple genes. There's a disease called Parkin, called PDR1. There is an accumulation of alpha-synuclein. DJ1 and PINK pathways are the one which controls in the familial form, the form that go from one generation to another. And the mutation in all of them start to lead some kind of disease, and that's a slightly complex pathway. I'm going to skip it and go towards the one that we are going to focus on. These are the symptoms of Parkinson. There is a motor symptom, that means where the body movement gets affected. In human patients, you will see slow postures, masked face, movement of hands become very difficult, and tremors in the lungs, and the gait, the child becomes very different. this is not only a neurological disease. We heard from Dr. Banerjee that any neurological disease would have some component in other part too. We heard about that it would have an impact on heart, there is an impact on lungs, there is an impact on brain, and then there's impact on liver as well. And as you see, the Parkinson affect almost all of those 5 vital organs too. Now, how are you going to research it? One way is you do a clinical research, which is being done, and we are also doing it. Second way is that we start setting up animal models. So we set up the models in mice and rat and could see how the Parkinsonian can be induced and changes can be done. And we have published that. But I'm not going to talk about this. I'm going to talk about very fresh data. And then we chose to work with invertebrates. Invertebrates there are a lot of model systems you can work with. So there is a worm called C. elegans, which is a transparent worm. There is a fruit fly, a Drosophila melanogaster. There are daphnias, which are very tiny ones in the liquid. And then you have sea urchins. You can use all of them if you want to. But what we chose to do, we chose to work on these two. And today I'm going to show a little bit of data on both of these models where we induce Parkinson's-like symptoms, and then we start to give them a various treatment and see the impact of that treatment of those model systems. So first, quickly we'll go to C. elegans, which is a very nice, beautiful worm. It's transparent. It's close to 1 millimeter in length. You can see by microscope. It has a very short lifespan, like 21 days, the whole cycle is over. Since it's transparent, it gives you a lot of advantage. You can generate a lot of genetic mutations, attach different kind of fluorophore next to the protein of your choice, and these worms will light up. You can see their colors. You can detect the type of changes you are looking for. And since this has been researched very well across the world, there are repository or different mutants that you can buy commercially. There's a center in US where you can buy them and start using them for the work as you need to do it. So we start working on them, and there are a lot of good insights you can get from them. And that's how they look. This is the photo from my laboratory that you can see the worm look beautiful. This is the anterior part of the worm, this is the posterior part of the worm, and they are very sensitive. If you touch them from the side, they actually change their movement better. And they can easily be bred, can easily be grown, do not require ethical approval issues like what you need for rat, mice, and guinea pigs and rabbits. And they feed on bacteria. So a normal laboratory which doesn't have large scope of operation can still house them and do a different kind of research. Now the query comes, why this and how they are relevant for neuroscience research. So in human body we have close to 37 trillion cells. C. elegans is just made of 959 cells. That's it. You can count each cell of it. And out of that, 302, more than 1/3 cells are neurons. That means 1/3 body of this animal or the worm is made up of the neurons. That means this becomes a good model system for playing for the neurological diseases. And out of those neurons, 20 are set near pharynx, which has its own nervous system. So basically the complexity of the brain system or the CNS, what we call in human system, exists in the worm as well. And you can see a comparative. How does it human system lies and what are the plain simpler way of comparing with the C. elegans. Now what we did, we started to do a modeling of Parkinson's in these worms, and we have multiple different kind of lines of these worms. So we have a normal worm which we call N2, which is a normal accepted name which has no mutation. Normal worms called N2. We work from another line called BG555, which is a strain of the worm where the dopamine transporter protein, PDAA, this has been linked with the GFP. That means wherever you have that protein, you will see green fluorescence coming out of it. And then we have a third line which is called NL590, where the synuclein have been attached with YFP, where you see yellow color. So basically, now we're going to play a little bit of colorful science, what we call in our laboratory bully, that you go with the colors and try to see how each thing getting changing up. And believe me, looking at colorful images of these worms is mind-boggling. And when you see them, you realize that you're doing the right kind of science. And we use inducer like 6-OHDA, which is 6-hydroxydopamine, which causes neurotoxicity, and then we look for how those changes are happening. Now this talks about so far what are the model system we are going to work with. How are we going to introduce or induce a Parkinson-like system in these worms? But what are the test articles? What are we going to test? The dawai kaun si hogi? तो दवाई ये. So this is the medicine that we work with, and the I think this guy is getting me. Give me a moment. This is the medicine we work with called Neurograde Gold, which has a combination. These are the combination mentioned there. Ekangi Ras, Moti Pishti, Rajat Bhasma, Basant Kusmakar Ras, Rasraj Ras, Jyotishmati seed extract, and Giloy stem extract. These are all classical medicines which have been defined in our ancient text for brain purposes. They are called Medha Rasayan. That means they are going to modulate the brain functionality. All we will combine them together, put them in a form of a capsule so that the patient compliance would be better. That means whoever is going to take it don't have to mix them and get them ready. They are now readily available, licensed by Ayush Ministry for clinical use, and they are now marketed. The Patanjali Mega Store in Varanasi could also have it. Now, this medication is actually defined for neurological problems. So we thought, why not we test this as such and see what kind of changes it can induce in the models that we are focusing on. And before we do it, this is one of the critical components as a biologist that I become very, very sensitive about. Like when we're making a drug from a natural source, there's going to be a lot of variants that nature is going to introduce. Like if I collect ashwagandha or giloy now, or the one I collect sometime in winters, or in sometime in summers, I might see a change. If I collect giloy from here, from Varanasi, or one from Bangalore or Hyderabad, I might see a change. And there are going to be a lot of natural variants. So there has to be a basic classification, that beyond that you cannot go through. That's what we call chemical fingerprinting. I need slightly more. Okay, I'll go very quickly. So Neurograde Gold, we did a detailed chemistry by doing HPLC and found what are the components we are looking for. Since it also contains some oily component coming from Jyotishmati, we look for the fatty acids, and by GCMS we could see what are the fatty acids contains. And now this comes a point that, does worms are happy with it. So we did a lot of experimentation for looking for acceptability of C. elegans by Neurograde, and as you can see, as the dose we went up to on all three different worms, but N2 versus 5901 or 555, the acceptability was good. Worms life was okay. Their progeny was also good. They are making their families the way they should be doing it. There was no change. But the good thing was that the lifespan of the worms which are mutant were lesser. So once you treat them with Neurograde Gold, you could see there was a very, very nice prolongation of the lifestyle. That means the lifespan of the worm which were getting decreased because of the disease-like symptom could also go back towards normal. You can then also look for the neurotoxicity which is introduced by 6-OHDA, which is happening on the normal worms. You can see the changes in their behavior, their chemotaxis, and how they are actually breeding through. Because the neurological functions also depend their behavioral changes, and the type of their movement, the changes, those can be also seen very quickly. And these are just the data that when you treat these worms with Neurograde Gold and later introduce them for the 6-OHDA, the disease-inducing phenotype what we see by neurotoxicity can also get reversed very well by these treatment too. And if you look for the chemotaxis, you can see the changes. You can set up the worms in a center of the plate and put a attractant on one time and triplant on the other side. Worms choose to go towards a specific path. They know where to go. They like certain things. I don't know why, but C. elegans seem to like ethanol a lot. So maybe there's some human correlation somewhere. But if you put ethanol in one side, C. elegans tend to go in that direction. But we didn't use it. We use butanol, and worms move in that direction. But if you introduce neurotoxicity, you would see their chemotaxis index becomes low, and that happens largely because of the total neurological shutdown process. And what is getting decreased, you see a very nice recovery when you treat them with Neurograde Gold. And then you can do microscopy and measure the type of degeneration that happens in the neurons, which happens because of the induction of neurotoxicity, and you see a good recovery when you treat them with Neurograde Gold. And this is a very critical slide too, because there you can see the synuclein imaging, the green color staining that you're seeing it in a normal uninduced worm versus the one which have been hit by NG, you would see the change are actually getting decreased. But as a control, we also use L-DOPA. L-DOPA is that drug which is there, is used in synthetic word in modern practice for people who are having neurological problem. This is also driven from a plant, and I saw a poster out there which talks about the same plant, what's called conch peach or kapi BHU, which extract from those seeds have been used. L-DOPA actually comes from the same seeds. And L-DOPA is being used, and you can see the response of L-DOPA and the response of Neurogrid Gold are actually very much similar, suggesting that there is a potential to see a comparative profile very well. And now, since I have a strong team that take care of the gene expression studies, so they look for various genes which controls the disease progression, and we look for multiple markers where you could see the type of changes that happens, and we could look for PINK1, PDR1 and CAT2, SAW2. These are oxidative stress markers, and their gene expression was modulated during the disease induction by 6-OHDA, and Neurogrid actually normalized all of those gene expression in the right path too. Now, this becomes a very quick summary of the work that we did on C. elegans, which got published in CNS Neuroscience and Therapeutics, the Wiley journal, which actually talked about that Neurogrid Gold has a potential to control dopaminergic neurodegeneration, Parkinson disease-like model. That's there in the public domain. One could download and read. Now, if ma'am you allow for 5 more minutes, I'll quickly show something about Drosophila, because I showed a picture and half of you might be thinking why you're showing and not talking about it. So we will request ma'am for 5 more minutes. So in those 5 minutes, I'm going to talk about Drosophila melanogaster and the model organism. It's a fruit fly, the fly that comes on your bananas or in your grapes when you keep them outside. Small little fly with the orange eyes. That grows in the laboratory very well too, and there are a lot of labs that works on it. We start using the same thing for Ayurveda as well, and try to see the type of changes you can induce. There's a good similarity of Drosophila with the human system, and I'll go very quickly to show the type of work we did. So what we did, we took a compound called MPTP, which is methyl phenyl tetrahydropyridine. This compound is also neurotoxic, and if you feed it to the Drosophila, they actually lead their behavior, which is actually normal. So the behavioral change on the flies can be measured by introducing this compound, and the assays are very simple. That there's something called climbing activity, that you take the flies, keep on a wire and you tap the wire. They will all go at the bottom, and you give them 2 minutes time and see how much they go to climb up. If they are having a neurological problems, they won't climb very high. So if you give them MPTP, you see as you increase the dose of MPTP, their climbing activity decreases, suggesting that these flies are going towards having a neurotoxicity. Now this becomes a very simple model. There you can then test Neurogrid Gold, which we did very promptly, and found that the climbing activity of the fly become normal. You can also look for the larval volume, because the flies has multiple things. There you see a good change, and the olfactory system too. But this is the slide I want you to focus. That the larva of the fly, if you keep it on upside down, it has a tendency to correct itself. It's like जैसे हम उल्टे लेटे हों तो अपने आप सीधे हो जाते हैं. ऐसे ही larva के अंदर होता है. If you keep the one ventral up, the larva will itself go towards the right direction and make the dorsal up. This capability of the larva is driven by the neuronal function. So if you give MPTP to these larvas, you would see their writing behavior also get compromised. And in those conditions, if you feed them Neurogrid Gold, you see a very nice recovery, suggesting that the behavior of the larva or the adult fly get affected very well. And these are just to show their activity on the crawling, but since man is looking me very strongly, so I'm going to go very quickly and take you this final slide, suggesting that if you look for the behavioral component, you can see the invertebrate model indeed has a capability to take the Ayurvedic drug validations. And we could see that either Drosophila or in C. elegans, we could actually generate the robust data which become acceptable by scientific fraternity and also by now regulatory bodies. We are generating a big dossier from invertebrate to vertebrate for various neurological changes, and most of those data is now available on the website of PRI, where you can see and download if you want to read it through. But what becomes more critical, which I come back to my original query about doing reverse pharmacology, is this, that becomes very critical for Ayurvedic drug discovery. We must do it with the right spirit, and if we go with the clinical control trials in parallel with this, you generate various level of clinical evidences combined with the preclinical validation, because there's certain things you cannot do with the human patients, but in animals you can do a lot of it. You can find out the target which are getting engaged, the target which are getting modulated, then it would generate a fantastic body of evidence which would be acceptable by people across the globe, and that we will take towards back to our mission, that we want to offer the world the science of medicine from the oldest medicinal knowledge base, Ayurveda, and it goes with the same thing, Sarve Bhavantu Sukhna, Sarve Santu Niramaya. With that I come to my final slide and go my acknowledgement to the team that has worked across. This is the team of PRF that you see over there, and we are blessed by Param Swati Acharji, Param Puj Swamiji, who give us our blessings on a daily basis. And what you see on the building, that's where we are. All of you who are happy to be in nearby, even 100 kilometers away, please detour a bit, try to go, come and see what we do, get feeling about it, and you can help us. We'll be very happy to take your help, or if we can help you, we'll be very happy to help you as well. You have been very kind audience. Thank you very much indeed. Thank you very much. And if there are any questions, I'll be very happy. I'm carrying a box of chocolates for the very good question you are going to ask. And if you do have, let me know. Or I need all of them. Hypernutrition. Any queries, folks?
Anyway, as usual, I mean, you might have done it on other higher order animal models, because the present physiological condition is different. And
of course.
So that actually doesn't fit 100% with this.
That's true.
So
Sir, that's also true with the human patient too. What might fit with the Indian patient may not fit the American patient, right? There are natural variants. So whatever data we generate, they all going to come with that rider, that this is not absolute truth. So whatever we are going to generate is just going to tell you, are we having a potential? And whatever we are doing it is being done for other medical system too. The rodent model, non-rodent model is being done for the pharma industry, and that becomes a path to go for first in human study, right? The clinical trial only start once we have in pharmacology and efficacy data and the ratio for pharmacokinetics generated in animals, right? Knowing it that whatever we see in animal may not all translate into human, but you must have the data. That's a regulatory requirement. So once you have that requirement for one mode of treatment, why not we have the same thing for Ayurveda? Right now, regulatory requirement in Ayurveda is not needed. You don't need to generate this for having a drug in the market. But if you generate this kind of data in animals or in cell lines, I have not shown a lot of data that we did with the human neurons in our cell lines. The type of the data that comes from there is mind-boggling too. You can see the pathways which are getting affected by the neurotoxicity and how the drugs are changing it. But having those data are actually good. It infuses confidence in the mind of the people who are seeing it, and the scientific fraternity start to accept Ayurveda as a medicinal system. And that's the action that we're going to work towards, and that's the motive that we have been working.
particularly for this neurotreat, I mean, this is very well sold in the market.
Right.
Many people are using.
Yes, sir.
So, pharmacokinetics has been completed on human.
Okay, now that's a very good question. So for most of the herbal drugs, doing a PK is an issue. Why? Because the compounds that you go to detect in the serum are tiny in nature. Because you are giving it, because your drugs are so diluted, you won't be able to measure it. But good thing about NeuroGrid Gold, that it has Rajat Bhasma, that means it has silver in it, which is a metal which can be tracked. So we have done this, and we have measured the PK in rats, PK in dogs, and PK in human are being done. So we are measuring that the drug goes to the right size. And the good thing is we can also look for the tissue distribution, because some people believe, pseudo-believe, that bhasmas have toxicity to kidney. Right? If you take it, then it's going to be a problem. That's what people think, which I strongly believe is a myth. If you do not manufacture bhasmas as per the classical formulation method, you will generate poison, which is true for making even paracetamol, the one you take at home. अगर उसको बनाने का तरीका ठीक नहीं होगा तो वो खराब ही करेगा. तो अगर सही तरीके से हम भस्म बनाते हैं तो वो कोई टॉक्सिसिटी नहीं दिखाती। न्यूरोग्रिड के केस में हमने ह्यूमन डोज के 28 गुना डोज तक 3 महीने तक हमने रैट्स को देखा और देखा कि बॉडी के कौन कौन से ऑर्गन में बायो एक्यूमुलेशन है। 33 के 33 ऑर्गन, 33 ऑर्गन्स, all have been harvested and sent for ICPMS for detection of additional amount of matter. So you won't believe, none came. Even the brain, no bio accumulation, which is considered to be the target organ. So we were generating those evidence to tell people that here is the data, don't believe us, believe the data. And if you want to test it yourself. And these are the guidelines that we have been following.
At least those components, are they crossing blood-brain barrier or that is also very important.
Right, sir. So out of these components, this silver is actually known to cross blood-brain barrier, but not up to the level which might become toxic. And the one what we see, this Basant Kusmaka Rus, this is also a fantastic drug for the brain, which has the gold in it. That's the reason the brand name is Neurogrid Gold. It contains gold plasma. And gold plasma is actually a good carrier for taking the drugs right into the brain. And we have done some radio level experiment too with gold plasma and also with ashwagandha, where we radio level them and give it to the rabbits. And after giving them, we measure for 3 hours constantly and see where you see radioactivity popping up. And we did Giloy in the parallel. Giloy, most of the thing goes to liver and the gut doesn't go beyond. Ashwagandha and gold goes up to the brain. Up to the 5% of ashwagandha was detected into the brain of the animal, suggesting that nature has something fancy. We have no idea how, but it does go to the brain.
Also it is important because you cannot expect everything to go to the brain.
It should not.
But so many things now they can take brain in their secondary level. Not necessarily everything has to go to every place. Biology doesn't come like mathematics.
Sir, I strongly believe that because in mathematics 1 plus 1 equal to always 2.
Yes.
In biology you can have a duplicate and triplet too. In case of cleanliness, thousands.
And I strongly believe that most of our Ayurveda formulations, they work on this. Not necessarily they reach to the target issue, but the content.
I think that's a very well said, sir. Let's clap for sir. Let's putting it so well. But one thing I like to also add, because I come from the pharma background, and most of my pharma brand friends pull my legs. कि कहाँ चले गए, क्या कर रहे हो, efficacy कहाँ से दिखाओगे? But the answer for that is that in modern pharma field, you are looking for a single target. You are engaging it with a very high affinity. Your compound goes to bind at a nanomolar range, and you take it forward saying, I'm going to modify disease. Ayurveda doesn't believe in that. Ayurveda says, I'm not going to have a single compound. I'm going to have 100 compounds. All of them going to have a loose binding to a given target. So what will happen? You will have a synergy of the effect. And that is the reason the side effect would be the low, because you're not engaging a single target with the high affinity. So off-target activity would be low, on-target activity would be diffuse, but would be more powerful. Any other queries from the people on the back who just woke up? Ma'am, please.
Sir, can you suggest some herbal which can work for neuroinflammation? Like it will support the neuroprotective form of microglia. So
The single herb that works very well for that, and we got some good data too, Jyotishmati, which is mentioned over there. These are the seeds, and the seed oil from that herb is fantastic and is very well defined in Ayurveda too. And for neuroinflammation, it works fantastic.
sir, it can be given orally or
It can be given orally without
intraperitoneally
IV. In human case, why would you go for IV?
for
Animals you can IV, no problem. But I think if you're going for animal study, try oral, because it would be connected with your future use if you want to go. IV will give you a better exposure, but per oral, PO is also equally good.
Okay, it can cross the blood-brain barrier with
Indeed.
Thank you.
Thank you. Yes, ma'am, please.
It was a very insightful lecture.
Excuse me, last question, because
Sir.
we are short of time.
Sure. Ma'am.
You can interact later on during the tea session.
No issues.
Ma'am, आप पूछ ही लीजिए.
Sir, how we decide the doses of the? There are so many ingredients in this neurobic formulation. So there are extracts as well. There are rasas as well. How we optimize the dose of the individual ingredients?
That's a very good question, and it's a very live question too. So what we do generally, we get inspired by the ancient documents, right? What is defined in classical texts. Most of these come from Bhavika Nightu. right? And some of them come from the book which talks about Raj, I think some book that talks about all the bhasmas. So there the human doses are very well defined. So that becomes our starting point. And from that point onwards, we make different stoichiometry, different combination, then we try which one works well in our cell models or in the animal models. On that basis, we will then take it forward. But whatever combination we take, we never ever cross whatever the doses formation given into our ancient text. Either we are working at that range or lower than that. And that seems to help. And there are a lot of patients who are taking these drugs and reporting clinical benefits. And it has been a really good result. Thank you. Sir, may I? Thank you. Thank you, folks. Clap for yourself too. Thank you.
Thank you so much, sir, for introducing us about the Patanjali Research Foundation and its role in about reverse pharmacology. Your perspective about the modeling Parkinson's disease in C. elegans and Drosophila completely changes our thinking in treatment of these neurological disorders. And I would like to invite Professor S.P. Singh, sir, to felicitate Dr. Anurag Vasu, sir. Without further delay, I would like to invite Professor Shur Pratap Singh, sir, a distinguished professor in the Department of Biochemistry at the Banaras Hindu University. Professor Singh's research focuses on the neuroprotective effects of traditional medicinal herbs such as Vidania somnifera, Tinospora cardifolia in the treatment of neurodegenerative diseases. With a PhD from IMS BHU and the postdoctoral training at the Johns Hopkins University, he brings deep expertise in the neural biochemistry, molecular biology, and the neurophysiology pathways. His work explores critical areas like neuroinflammation, mitochondrial dysfunctions, and the herbal-based therapeutics interventions, which bridging traditional knowledge with cutting-edge science. A strong advocate of interdisciplinary research, Professor Singh has contributed significantly to the field through impactful publications and ongoing collaborative projects. Please join me in warmly welcoming Professor S.P. Singh, sir, with a huge round of applause.
Thank you very much for this huge introduction. And I was really impressed by the previous speaker. And by chance, this is back to back his Parkinson. And so today I will talk about validation of all those drugs like medicines, urban medicines, which has been used in several neurodegenerative disorders, and one by one, each drug, each medicine has been validated by one single PhD student. So several students and several herbs, and we have really validated on very good result, and kind of experimental scientist having several knockouts and other kind of work. But when I returned India, I just met one of the very famous Ayurvedic physician, Mr. Ram Singh, who was a chickisha. So he said, all research are not good for India. So you better not make any knockout or any crazy flies. So you should rather work on validating what we provide in the clinics, and patient get treated and we are all happy. So, but we don't know how it works and where it works. So that was the goal I started in 2008 and now it's like 45. Since then I am working. Okay, so this is the neuroprotective potential of Indian medicinal plants and their bioactive compounds against Parkinson's disease. So, little bit yoga and Ayurveda. So, yoga, since I have carried away 2 DST projects on yoga and intervention in amyotrophic lateral sclerosis, ALS, and yoga, and we have got a positive result from MRI data, and which is going to be published very soon with the collaboration of one of the neurologists in our hospital. So that's one. And another, in the psychiatric department, another psychiatrist, I got a DST project, which is intervention of yoga in schizophrenia. So this is ongoing one, and the other one is over. So with this, I know yoga, many people they give very different definition, but as Professor Rao was telling you during his inaugural session, that yoga is nothing but is a way of life. The way you interact, so that creates a problem. So this is neurotransmitter balance. So it's a way of, even if you are driving a car and a red light, you just keep on honking. That's it. So, every reaction, every in your action should be yogic way. So that's it. Otherwise, Patanjali has given like a 5 parts of yoga, yama, niyama, asana, pranayama, dhyan, dharana, samadhi. So all those things are there, but in a short, one sentence, it's a way of life. The way you live and react with environment and whatever. Okay, with this in background, let us talk about Ayurveda, which is nothing but wisdom of life. It's a deeper level of comprehension of bodily tissue and energy. And this is the 17th incarnation of Vishnu, or Vishnu. And it's called, God of Yoga, Ayurveda, which is Dharmantam. So it's ancient medical wisdom from 2000, 5000 years. How it treats Ayurveda? Holistic approach to achieve optimum health. So our goal is to achieve optimum health. And optimum health, and by achieving optimum health, agelessness. Enlightened. That's the whole purpose of this exercise. As you know, our body is made up of Panch Tattva, right? It's a Jal, Prithvi, fire, Agni, right? And Akash and Vayu, Panch Tattva. That's Panch Tattva mein vileen ho gaye. So these are the things. And Ayurveda believes on three things: Kapha, Pitta and Vayu, the balance of these three. So as long as it is a balance, you are healthy, you will be healthy. There is a disbalance, any of the thing, you get sick. So that is the principle I am talking. So now Ayurveda and neurodegeneration. There is a term rasayanas, which means a vital nourishment, and they are also called mental elixirs. What they do, they promote health, delays aging, and strengthen tissues. they have 100 times more powerful, they are 100 times more powerful antioxidant agents than our conventional antioxidant. And sorry, some of these you see our wonder land, ashwagandha. Dr. Vaishnav has been mentioned many times. Muleti, Brahmi, Giloy. Giloy is nothing but Pinospora cardifolia. So you just know. This is the second, since my laboratory is working on Parkinson's disease, and it is known to be second most common neurodegenerative disease after Alzheimer. And the symptoms are tremor at rest, rigidity, depression, anxiety. So say it's a motor symptoms as told, motor and non-motor symptoms. So people have been focusing on motor symptoms more because this is a visible symptoms, and by the time you come with the motor symptoms, slow walking, tremors and all that to the clinic, your 60 to 70% dopaminergic neurons are gone. So it starts way before that in the form of non-motor symptoms, constipation, and say depression, anxiety, gastrointestinal troubles, dementia, all these. And there are several. So degeneration of the dopaminergic neurons in substantia nigra, particular part of the brain called substantia nigra in the middle brain, middle part of the brain, is the Parkinson's disease. And see, here's the picture. So this is substantia nigra. And though it was well known into the modern world after James Parkinson gave the name Parkinson. However, in India, in Ayurveda, it has been described as a kampvat, 5,000 bag. And these physicians is treated this patient with ashwagandha, concoction, which is mucunabulase. So this is again the same thing, and you all know that onset, you have already. And this is a picture like this: the loss of memory. This is healthy brain, and this is a sketch of Parkinson. You see this loss of dopaminergic neurons here, and loss of memory. And this is normal neuron. You see again the history. I don't want to get into this. And prevalence also has been told to you. We will stop. What are non-motor center? I just was mentioning here, but there are many others. You can just quickly go through these images and you can find out these are not. So, pathophysiology of Parkinson, as per there is a Ross generation, there is a neuroinflammation, there is apoptotic death, there are several. So I will come one by one. See, degenerating neurons, they produce reactive oxygen species, right? And which leads to oxidative stress, which leads to microglial activation and reactive astrocytes. In turn, there is a production of pro-inflammatory mediators such as TNF-alpha, IL-1 beta, and 8, etc. And finally, degenerative neurons. So, this is, of course, Parkinson is of like two kinds. One is sporadic and another is genetic. And Dr. Sab has already shown you several genes involved in Parkinson. And but our model is like a sporadic model, environmental induced, chemical induced model. So there are several chemicals which produces Parkinson, and most of them they impair your electron transport chain mitochondria, and hence the generation of data. So these are like MPTP, very popular, but it has some limitations because there is no new body formation. Rotenone is, I would say, one of the best model because this is a acute model though, and rotenone is the chronic model. And Parkinson is a chronic disease that slowly, gradually builds up and lost. It's not like 60% neuron will be lost in one day. No. So that's why this we have tried all those models in our lab, and finally we stick to this rotenone. And sometimes back and forth like MPTP, depending upon what we require. So current therapy in neurology, if you talk, these are the drugs. The most common drug is Syndopa, right? It is a combination of L-dopa, levodopa, and carbidopa. Why? Because if you give only L-dopa to the patient, it's a emetic agent. Patient will start vomiting and all those. There will be a lot of peripheral problems. And why carbidopa? Because it is a decarboxylase. And this L-dopa is converted to dopamine peripherally because decarboxylase enzyme is everywhere. So immediately in the blood, the moment you eat and absorb and goes into the blood, it's converted into dopamine. And dopamine creates problem, right? And this is in peripheral circulation. So therefore, this Syndopa is given, and which is a combination of both, and it inhibits this. And once it crosses the blood-brain barrier, goes into the brain, then it is done. Converted into dopamine and works. And the latest one is DBS, deep brain stimulation. So in India also it started. So you put electrode inside and keep on stimulating your dopaminergic neurons and hence the release of dopamine. But they possess solitary empathic advantage, no effect on the curbing the disease progression with numerous side effects. Because those persons who has been on these drugs, after certain time, like after certain 10 years, they have a very bad condition, that will be very bad. And sometimes they die also. So that's why I would suggest to go for Ayurvedic medicinal plants, Ayurvedic drugs. So it's an ancient Indian medicinal system practiced for 5,000 years, as I told before. And a course of purification, the guidelines. Ayurveda encompasses a wide range of treatment, including guidelines for healthy lifestyle and diet. That's again, healthy lifestyle and diet. Second, a course of purification to detoxify the body and energize its healing abilities. In this, during the lunch this morning, one of the speaker was discussing with me. My son is like in IIT Delhi, and their lifestyle is very different. All night they will study, they go to bed like 4 o'clock in the morning. So we should think of something for these kids. Even my daughter, when she was doing MBA, I am in Ahmedabad, so every time at 4 o'clock, okay, I'm going to sleep, go to sleep. This is kind of the situation. So lifestyle is, for them, lifestyle is very different. And that is the cause. We should definitely work on it and do like all premium institutions, we should think of doing what should be their lifestyle. They are brilliant kids, and they are top institutions in India. But they are very stressful right now. So majority of Parkinson patients are treated with medications to relieve the symptoms of the disease. None of it stops. Progression can stops with all disease. I'm going back. Okay. So Ayurveda and Parkinson disease. Here you go. Kapil Kapish, which is mucinephritis, and Dr. Saab has named something different you call it. Moj. We call it Kemaj. Yeah. Moj कहते हैं क्या? Ayurvedic physician here है, तो he can validate that. तो Kemaj बोलते हैं. और Bami, देखो commonly we have worked on it. Every curcumin longa, ashwagandha, vidania, somnifera, garlic, and insulinose. So all these have their positive effects. So now I will show you some of the data, lab data on this, which has been already published and well established. Quinquinaprine extract eliminates amphetamine induced neurotoxicity by NF-kappa-B and phospho-AKT. So we have gone to the pathway, how it is alleviating the symptoms, right? So, and we have, the way we have done, so any extract, normally we take extract, right? And when some of the people they say, oh, this is like having so many components, we don't know which is causing what, like pharmaceutics people do. Okay, no, it has with furin, it has biphenoid A, without B and whatnot. So for them, we went for some of the component also, and I have the result also. So, which is in, so normally what we do, we create a model and preferably rotinol, and then we prove our model that it is a Parkinson, showing a Parkinsonian behavior, and we won't call it Parkinson. Because Parkinson disease cannot be produced in animals. It is a human disease. You cannot produce. So then that's why we choose them. Parkinsonian symptoms. So they will have slow movement. They will have slow movement. They will have bradykinesia. They will have tremors and all those. So in animal like mice, we gave this rotinol and we chose like 3-4 groups: control, metal control, positive control, and then we like a disease mice we treated with rotinol, and then these drugs, and so like what is happening. So you will find the middle one in this case is PD, in Parkinsonian, where we have treated with the rotinol. And we look at their behavior parameters like standard techniques, hanging test, narrow beam walk, and several others. Rotenrod is one of the very popular in pharmaceutical students, like Rotenrod test, with moving rod and how long it can stay on that. So they will say this is a control one, and they can stay longer duration on the rotor moving rod. And here is the Parkinson's animal. And when we treat with mucinephritis, look at this, they'll stay cut up. And then similar thing hanging. So we put on like a cage, turn it upside down, like a cage is like this, turn it upside down, and put our animal here. So they can hang longer duration, normal, for those who have made Parkinson's. So hanging time. See, it is reduced in PD, and again the treatment will come up closer to the normal. And this is like a narrow beam walk. We used to allow them to run on a narrow beam of scale, and then you see the control one go faster, quickly they will, because mice they run quick. And when you treat them, put it on, it's bleeding, so they will go slowly. So they will take longer time to walk on a narrow beam as compared to the control. And when you treat with here your Viquina, see this is better. So likewise, so first we have behavioral test, then we go for biochemical, several biochemical tests like catalase, nitrite, NPO, GSH, several. And then we go for directly cutting out the brain, cryosectioning, and staining with dopaminergic neurons and showing that yes, they have gone down when you make practice. But very few dopaminergic neurons are there as compared to the control. And when you treat, they are light up and they have better number. So, and this is like we did like a SPLC analysis, component analysis, all those things for those who created questions and they are not convinced. Of course we have to convince. And this is all those immunofluorescence data, immunohistochemistry of the brain, direct visualization. So this is this panel shows NF kappa B. So the NF kappa B is nothing but inflammatory marker. You see this is PD, it goes up. This is control, this is PD, this is pituitary. See? And this is TNF alpha, again inflammatory marker. This increase in case of PD, goes down. And this one is ICAM, again marker of inflammation. And this is so hard to read. I have to use my glasses. Anyway, ICAM and something INOS. So all those inflammatory markers were increased. You see this panel, middle panel. Interferon gamma? I can't. Okay, here I can see, yes. So these are some of the previous workers, like I think Sajidanand also. And then we looked at several, this is amphitic treatment and with muconapirins, you see this amphitic treatment. So this is like a GFLP. Okay. GFLP is a remarkable again, and then that is a transporter, and then this is a pH. Tyrosine hydroxylase is the neuron, is the precursor of dopamine, which is formed in the dopaminergic neurons. So, with tyrosine is a rate limiting step of making dopamine. So, we have antibody against pH, tyrosine hydroxylase. That's how we identify dopaminergic neurons. Okay? And this is like a nuclear. This is another further mechanism, is a nuclear translocation of NF-kappa. NF-kappa has a two subunit, so it breaks and goes to the nucleus. So this came in 20 years in aging neuroscience. I think Sajidanand was one of the PhD students, and then another student came with idea of TNOs. Let us try this test. You will find the similar result. And here we have two bars because one is control and another is impossible. So vehicle control and is impossible. And this is like a PD. And then the clinic. Similar story. And these are some of the protein direct western blot and other things. And of course, real-time PCR also for inflammatory markers. See, we are biochemists and molecular biologists. So we have all those techniques in our hands. Anytime we can apply those. So this came, and this was the MPTP model. against organum. BPA protects against amphetamine intoxicated neuroinflammation in Parkinson disease through NF kappa beta and phospho-AKT signaling pathway. So we came out with the pathway. Then Neospora cardifolia suppresses neuroinflammation in Parkinson mouse model. And this is, did I show you this? Okay, so here is GFAP and IBA1. IBA1 is a marker of microglia. So some reviewer asked, okay, we saw the microglia is, what is the situation of microglia in MPTP treated? So you see the picture. So again, TNF alpha and alpha. So this work, again we published in Neuromolecular Medicine. And then one of the students worked on Varanasi. So this is, we combined with two works. One of the work in my laboratory, some of the students were working on BPA. It is a component of plastic. Any plastic bottle or anything, it has BPA protein. You have the thermal paper of, print out of the ATM machine or anywhere you have the print out, it's all BPA. We have tested, we have done with HPLC and BPA is also. So BPA is such a harmful compound that we have shown in one of the places we work that it causes male infertility. Those pups which are treated with BPA, they cannot produce babies as opposed to control. So we wanted to see that if there is any effect of BPA on the brain and if there is something that can elevate it. So this is yes, answer is yes. Neuroprotective predominania somnifera in BPA induced cognitive dysfunction and oxidative symptoms. And then we came for individual compound now, orsolic acid by HPLC, HPLC and all those things. We figured it out. Okay, Lupina has quercetin, Lupina has orsolic acid, Lupina has other things. So one we do, orsolic acid. No question from any pharmaceutics person. Now it is single compound, not chlorine, orsolic acid. And the result is the similar. I don't want to go and explain the similar things over and over. So we showed that neuroprotection of rotenone induced Parkinsonism by orsolic acid in PD. Am I going beyond the time? Or is I'm in time? Okay. So I will be So neuroprotection of rotenone induced Parkinsonism by orsolic acid in PD. And then again, chlorogenic acid, one of the components of green coffee. And we have shown that chlorogenic acid attenuates mitochondrial mediated apoptosis senescence of the neurons. So these are some of the data showing chlorogenic acid suppresses amphetamine-induced apoptosis via AKT, GSP3 beta at 1-2 signaling pathway in Parkinson's mice. So again, similar thing. So we published in neuroprotective effect of chlorogenic acid on mitochondrial dysfunction mediated apoptotic death of the monogen neurons in Parkinson's model. So in summary of this study, I would like to explain you that when there is an oxidative stress, so all these cytosolic superoxide dismutase, then mitochondrial dysfunction, GSH, electron transport chain complex 4, 5, 1, 4 and 5, they are all down, goes down, you see. And in case of oxidative stress, when there is oxidative stress, and LPO and nitrogen oxide goes up. Okay? And neuroinflammation, neuroinflammatory response goes up, like NF-gamma beta, L1 beta, NF-alpha, osteoporosis, all these. INOS is increased, ITEL is right. Then, and the phenotype here you see, motor impairment, reduced survival, loss of dopaminergic and nerve tracking. And mitochondrial dysfunction mediated protein cascade also occur when there is oxidative stress. So BAX is increased, caspase 3 is increased, PCL2 and phosphoAKD1, ERK1 and phosphoGP1, they are all down. So this is the molecular mechanism how it's working, and the moment you use these Ayurvedic herbs, you see what happens. It is blocked. This step is blocked. This is horrible. So this is the effect of these drugs. And this is my team. Most of them are in US. Most of Bhalia is in Louisiana State University. Soumitra is in Johns Hopkins. Chetan is Russia. Birla is in New Jersey. And he's in Houston. And then this is Hajra. Hajra is in here, Roorkee. This is in Roorkee. Sorry. Hajra is in Roorkee. Aina is in New York. Albert Einstein. So most of them, none of them are here, except Rudy, I live closer to you. So this is a very good student. And sir knows all these students because they have been presenting in professor. So I'm happy that only one girl is left here. She also got a job and she's a forensic officer. But she has taken leave and she'll come back and finish after 3 years. She'll just come back. So that's it. And this is like, aerial view of our university with the temple in the middle. So we are in a very unique situation, and you can identify somewhere we are here. This is IIT. Now IIT. And main gate. So source of inspiration is here. The moment we get into the gate, we get like energized. And हमारे समय में बच्चे जब हम लोग थे स्टूडेंट, because I have studied 14 years here. So जब हम लोग थे तो कहते थे कि मालूम जी छड़ी क्यों लिए, बिना डिग्री लिए बाहर निकले तो डंडा मार के अंदर चले जाओ मार. So this was the we used to talk about degree लेके निकलना चाहिए. So kind of and some of the foreigner, like foreign visitors, my mentors from Tokyo, Chicago, they came, they all mentioned that Surya, this your university is city within city. This is a different, perception. Outside the gate, boundary wall is a different world. The moment you enter, 4 degrees down, temperature is 4 degrees down. Most of the time it is raining, but outside is fine. Anyway, this is all about our monitor. I'll be happy to take any questions if you have. Anyway, I'll be available here. Good.
So most of your formulations or whatever drug you have used, they are as an antioxidant, right? So in what sense it is different to the normally available vitamin C, vitamin E? They may have
100 times
similar
better than synthetic. That's what I told you. That's the benefit of iron.
So it means they're pre-exposure or pre-supplementation.
So we always give like a pre-treatment with this and then MPTP or whatever.
Pre and post.
Yeah. 1 week before we start this treatment and then together.
Immediately you get a better response.
Because it goes with the treatment. Whole 3 weeks is good. Rotenone plus liquidum, like this. Pre-treatment for a week and then continue with the agent, chemical
So it
agent.
doesn't hamper your model generation of the model.
I have proven the model. The very first thing is prove your model. Unless people will question like you have. So that's why we have shown the behavior, biochemically, and direct visualization of the model. What else you need? It's an animal. You cannot say anything. But you can get brain from animal. That's why we work on animal, and that's why it also started working on cheaper model. This is price, no price, see elegance, very few genes. Right? I also intend, several senior people, they suggested me, why don't you go on this? This is an expensive model, a lot of ethics. So, but then, somehow we could not develop the silly elegance. Because this is like, now this is time to retire. 2 more years, that's it. Anyway.
Thank you so much, sir, for explaining us about the role of medicinal herbs on the treatment of Parkinson's disease. Your talk offered a clear and valuable perspectives on blending traditional remedies with modern science. Now I would like to request Professor M.K. Thakur to felicitate Professor Sur Pratap Singh, sir.
Kind of an inspiration source.
Still is a pressure to all.
Even now, of course.
Moving ahead, I would like to welcome Professor J.S. Tripathi, sir. He is a senior professor in the Department of Kaya Chikitsa, Ayurvedic Internal Medicine at the Institute of Medical Sciences, Banaras Hindu University, Varanasi. With a distinguished academic and a clinical career, he served as the head of the department from the 2016 to 2019 and has played a pivotal role in advancing the integration of classical Ayurvedic knowledge with contemporary clinical practices. His areas of specialization include neurodegenerative diseases, chronic systematic illness, with a strong emphasis on the holistic, personalized Ayurvedic management. Professor Tripathi is also recognized for his contribution to Ayurvedic neuropsychiatry and lifestyle disorders, and he has mentored numerous postgraduate and the PhD scholars. In addition to his academic contributions, he actively participates in national and international seminars and continues to provide expert consultations at the BHU Ayurvedic Hospital. His work reflects a deep commitment to preserving the essence of traditional Ayurveda while promoting evidence-based approaches in the teaching, research and the clinical practice. Let's welcome him with a huge round of applause.
Thank you and good afternoon everybody. As told by the organizers, we are running short of time and already previous lectures we have. So I will try to be a little quick and we can discuss many things later. But my sitting in front, Professor M.K. Thakur sir, the President of SNCI, and our inspiration at in the neurosciences at Varanasi University. Dr. Anurag Varshney, whom though we belong to India, but I met first time in the South Korea in 2023. I and he met in the South Korea there. We stayed for 1 week there, 1 week. And Dr. S.P. Singh sir is a renowned person, and we have a good kind of relationship, research relationship. And I work one of my in the research project we have on the Brahmikit we worked on the Alzheimer's model. This was very good study, and the Brahmikit has got a very potential into the neurogenesis, especially the Alzheimer's. So that was the very important area we worked on, and other persons from faculty members, Dr. Sanak, Dr. Vandana, and Dr. Srilata, and all those the distinguished scientists and the students who are there. My topic is a little bit different because I've been talking about the non-pharmacological approach. We have been learning since morning with the pharmacological approach, and that too from the core scientists. I do not belong to the core science background. I'm a clinician. As I told my introduction, we have been working in the hospital especially in the area of neurodegenerative and neuropsychiatric problems. We are doing with, and since last several decades. But we have a collaborative study. We develop certain insight regarding with the animal models and those things. So I will be talking about on the meditation, because similar to the what we can get from the herbal drugs, and how they can bring about so many changes into the brain and neuroscience. Similar changes can be brought with the science. The researchers have told that many changes can be the neurotransmitter changes and all kinds of the changes with brain wave activity. All we can find with the meditative practices as well. So that can be my topic is just to sensitize and just to orient the new generation of the scientists, body scientists, that some of them they can choose this area also, because this has also potential to work and to potential to prove yourself that this can work very well. So that is why I'll be talking about meditation as a neurotherapeutic tool and the bridging the brain science and psychiatry, because since neuropsychiatry is my area of study, you bridge the brain science and how the meditation is a tool we know is from ancient practice, and it's our own, and as in the morning sir was talking about Indian knowledge system, so belongs to one of the potential areas in the Indian knowledge system, and it has kind of meditation is such one word, but it has got so many different kinds of orientation and different kinds of meditations are there, but they are working into the similar fashion in the neurochemical effect, and the changes into the hormonal system in the body is in the more or less in the similar fashion. But as we did the meditations are in the technique where attention is brought on the either in the breath or on different kinds of mantras or different kind of bodily sensations. We used to focus on our attention, and the purpose is different. Ultimate purpose is to get the ultimate sleep, but it ranges from stress reduction, increased calmness, and enhanced overall well-being, and self-awareness is the main motto of the institution. So before that, it's nice to that what is the basic origin of the meditation. Meditation though has got basic origin from different kind of our Upanishads, our Vedas. There are all kinds of the traditional system of the meditative practices are there. But ultimately, mostly we talk about this Patanjali Yoga Sutra, which is called where they define the Tatraik Tanata Dhyana. It's very important. From there, all kinds of we develop this. So Tatraik Tanata is the continuous, uninterrupted flow of mind towards the object of focus. Towards the object of focus, we continually our attention is at one point, one object of focus. That is the meditation, and there are so many kinds of meditation which are in vogue practice. Many of you you may not be knowing. Similarly, in the Granth Samhita also it says that meditation can be done on three forms. There can be a stool form. The stool form are the physical form. Sometimes you can meditate on something physically. You can see the visual practices. There can be on the Jyoti, the meditation, brilliant lights can be there, like in the Tatak activity we do, the meditation, and the Sukshma meditation on the formless Brahman or some kind of, any kind of deity you are following, your Ishtadeva or something, you can do the meditation on that also. So it can be of three types broadly, and from modern understanding we can divide that meditation can be focus meditation or concentrative meditation, which will be on with thought, any kind of thought, any kind of sound or visualization of the breath, mantra or candle flame. This can be there. And then it can include the breath awareness or visualization. Visualization is one important potent thing which we have got. Everybody has got, because our mind does not know that the difference between the visualization and the real reality. What is the reality and visualization? The mind doesn't know the difference, because many times if we go to sleep and we get nightmares, we wake up with the sweating and then kind of increase the heart rate, elevating, increase respiratory rate. All these are similar to that, just in the sleep stage. You are moving here and there, and somebody you got a frightening scene. So similar to frightening scene you see in the night. So in the living time, that also will be similar. So this is very important, that what you visualize is automatically reflected as a original thing. So the focus attention, the open monitoring, or sometimes this is called mindful meditation. This is extremely common. Whatever I am talking about is the scientific evidences related to the mindfulness meditation, which is on the focusing without any attachment on the thoughts or feelings, whatever we get, that focus our attention there, so that we'll get mindfulness. This is very common nowadays and practiced, and so many researches are conducted on the and the most of the researches they are conducted on the mindfulness meditation are in the Western world, in the transcendental meditation. These are extremely common, and then there can be automatic self-transcending type of meditation, and sometimes the meditation, the earlier stages can be done with guided meditation, where somebody is there, either a person, audio or video, to guide you to meditate on something in the initial stages sexually. So common meditative technique, I will not go into the details, but one of this most important transcendental meditation, because why it is important? Because the first time when the study on the meditation started in 1950s, the first person was the Herbert Benson, who did in the Harvard University. He was working on a stress model, how the stress creates a different kind of bodily changes into the our different parts of the body, and how your cardiac function changes, how the respiratory function, and what are all kinds of the physiological changes, all kinds of biochemical changes that are brought about by the stress we see. So all those can, while working on the transcendental meditation, he found that whatever the changes are found by are done by the stress, they all can be reversed when we use the meditative practices. Means the stress, all those physiological functions, all the biochemical changes, all the hormonal changes which are produced by the stress, they can be reversed. Because Dr. Anurag was talking about reverse pharmacology. This can can be reversed by when you use the transcendental meditation. So where the person got interested to the understanding meditation, and then researches and meditation started. So this was the pioneer work of him. That's why the stress creates the stress response. Similarly, the transcendental meditation creates the relaxation response. The word relaxation response was coined in there from the 1950 by the work of Herbert Benson in the Harvard, which created all those things. Because the word is the accepting yoga, not because of the government effort, because so many researches have been conducted and people have believed that all these practices have got a scientific value, and they have. So that is why it's not because of the emotions, out of the Indian knowledge system, that word is accepted, that we are accepting World Yoga Day, and United Nations universally accepted this thing because of the scientific evidences which have been created later on after the work of Herbert Benson there. So the transcendental meditation was most popular at that time, and you can get the centers all over the Europe and the major American cities there. Founded by the Mahesh Yogi, and it works on these certain kind of the mantras there, the mantras which are involved into the transcendental meditation. Then loving kindness meditation is another one. Vipassana meditation is also very important. From the Buddhist tradition from India, it developed, which focuses on the bodily sensations and also thoughts of impermanence are just put into the meditators. Then the mantra meditation and Zen meditation is also a kind of Buddhist meditation which have a special focus on the breath and the thoughts without attachment, just like a mindfulness meditation. Now this we are talking about with the herbs and so many chemicals and the dust, we are bringing about the neurotransmitter changes. But all these neurotransmitter changes can be have been found to be chronic meditators who have meditated for the few months to years. They have been found these studies have been conducted on their brains and these persons. So because one of the limited neurotransmitters is the GABA, so it increases the meditation increases the GABA in the brain, and it also has that is the effect on the reducing the anxiety, improved mood, and a stronger sense of the calmness. Then the serotonin also is shown to boost meditation with the meditation, and so it causes leads to good kind of mood and improvement into the elevation of the mood and the happiness, which is seen into the depressed state. The mild to moderate kind of depression is there and effective. So it has got antidepressant effect. Similarly, dopamine we are touched upon is a biochemical which is responsible for the motor activity. Just we heard last 2 lectures. Also from the is responsible for the pleasure, the learning. And it is elevated with the meditative practices. It has been found in these studies. Also the cortisol level, which is elevated into the stress condition, that is shown to meditation has been shown to lower the cortisol level, and also reduces the relaxation response because of this one. So this it's not only that there are certain the biochemical changes are observed, but many times the structural changes also seen with the in the brain part in the persons who are meditating for a long time. Their brain, scanning of data, the brain imaging has shown that we all know that this hypothalamus is crucial for maintaining the physiological homeostasis, as important for mediating the stress, emotion, and the cognitive functions, and also the for internal physiological states and sleep-wake cycle also there. So the certain meditative practices have found to influence the hypothalamic activity by modulating the parasympathetic nervous system, and this HPA axis, which is the hypothalamo-pituitary axis, which is the main stress regulating system, that is also to be down regulated with this with the meditating persons. And the neuroimaging has shown that the enhanced hypothalamic function and contributing to emotional regulation and improved sleep and overall cognitive physiological well-being are also shown to be improved into the persons who are especially into the mindfulness meditation we are talking. Studies conducted. We are all aware that brain generates like the ECG, we are telling EEG, the electrical activity of the brain generates EEG, and there different kind of waves has been identified. For the students, because the beta waves is the normal our activity, and in the anxious state, the brain generates beta waves in a mode. But in the relaxed condition, when awareness is there, then the alpha waves are generated. After that, the delta waves and theta waves are generated in a more meditative person, in a more cool and calm person. The theta waves are generated in a deep meditative state, and the gamma is an heightened perception. So, yeah, it has been found that meditation changes state of relaxation, and it produces a more of the alpha theta wave activity was found in persons who are meditating. So alpha automatically brain goes to relaxation state, and then theta wave activities and alpha activities are become very important there. Long-term meditation practice can make alpha wave the dominant brain activity. This has been found, and theta wave activity also not only the increase into the alpha wave, but the decrease in beta wave, which is increase into the anxious state, in the stress state, that is also seen. And in the deep meditators, the increase in theta wave activities has been shown into the EEG in the finding. So this all proves that, and above all, we all know that the neuroplasticity is one of the remarkable ability of the brain, that how it has got the ability to change, reorganize itself and rewire its neural connections in relation to the new learning experience we have, and the environmental changes also. The brain has got capacity of neuroplasticity, and because of this, the brain has got capacity to form new neural pathways into there, and strengthen the neural connections and generate new neurons, and enabling the improved function is there. So neuroplasticity is one of the important capability of the brain, and it produces, because of neuroplastic activity, it produces many kind of functional changes and the structural changes also. Functional changes are rerouting the brain functions from the damaged area to the healthy ones. That is why we get improvement in many of the rehabilitation. Many of the patients who develop the paralysis and all those, and degeneration, neurodegeneration, we can get it depends upon the person to person. The level of neuroplasticity depends that to what extent the person will have the improvement in this condition. Similarly, there is structural changes also found in the physical shape and the connectivity of the brain tissue. This is also important in those functions. So the major mechanisms are this important, because all these are affected by the meditation. If you go to, because I will not be talking in detail, but if you are interested, you can go to the researches conducted in there, and you will find that the synaptic strengthening and pruning, this can be the synapse, the strengthening of the synaptic activities, and the pruning also. Pruning means those neural pathways which the brain is not using. Something with the redundant pathways, all that they can be pruned just like a shrubs in there. So there can be destroying those pathways and using the newer pathways. This can be done. Axonal sprouting, growth of the nerve endings can be there. It was believed that the neurons cannot be regenerated, but the work of the Ashwagandha work in the Japan, it is used that detected dendrites can be regenerated there. So the regeneration can be possible. It's not that we have got the belief that the neurons cannot be regenerated, but they can be regenerated to some extent if they have got this neuroplastic activity. And with the work on the Methanian somnifera just there, first time we got that the neurons can be regenerated. This was this question. Then the cortical remapping, this is done by this activity. So and this kind of neuroplasticity play an important role into the brain injuries, knee stroke, and the sensory loss, all those where we find that this there. And the studies have shown, especially on the mindfulness meditation and the neuroplasticity, that it increases the cortical thickness. With the neuroimaging technique, that it's not only the biochemical changes, that functional changes, there is many structural changes can be has been seen, and repeatedly, not only single study. Several studies have been conducted. Increases cortical thickness, mainly the prefrontal cortex, which is our responsible for our major executive functions of ourselves, and then the anterior cingulate cortex, which is also a part of this executive function in the body, and it also reduces the size and the activity of the amygdala. Amygdala, this part of this limbic system is related with the stress response in the body, because the reduction to the amygdala and its reactivity changes, so persons have got improved emotional resilience and less ability to just respond in case of the stress conditions. Similarly, the enhanced connectivity has been observed into these areas. Prefrontal cortex and default mode network, DMN, is another important network in the brain, which is responsible, which is activated when we are in this where we are not working, where temperature DMN is activated, which is responsible for our very important for our cognitive improvement, cognitive well-being, our emotional well-being. The DMN the network is extremely important. That is also shown to be enhanced connectivity is seen between the prefrontal cortex and the DMN with the persons there. And also it reduces ruminations linked to the anxiety and depression. Because many of the things we found that similar kinds of thoughts come to the person. Many patients will come in the negative all the time, they're thinking about negative things, and this is called the ruminations. This is called the ruminating in a different time without wanting somebody else, got different negative kinds of thoughts and certain thoughts which are not desirable by him. So those things, this can be these are reduced because of this improved connectivity which has been seen. Also, this is a 40 days study on the important study which is published in this one. This also shows that the left precuneus and left superior parietal cortex, their thickness is improved. So this also shows that left precuneus is also important part which is related to the cognitive ability and cognitive flexibility. So this also leads to it confirms the cortical thickening associated with the meditation, which is very important in long-term executive functions. We have already discussed so many structural changes. I just skip on this one. DMN also we would just did this one. The most important part of being a human being, cognitive function is the executive control circuit, and this is made up of the DMN and the central executive network which is there. It has been demonstrated by several studies by Brewer et al. That experience in meditators with the less activity is major DMN regions has been observed, and the Tang et al. Proved that the meditation modulates the core neural network and auto enhances this executive function and self-monitoring through network reconfiguration. Because that is why the better activity is there. The person able to work in a better way, in a more skilled way. Those who are meditating, so this can there. Also, increased activity has been observed in DMN, SNCI, and the other areas of the brain. This is important. And meditation also enhances the functional connectivity in the DMN and the executive brain function. So these are all kinds of this structural and functional changes with the meditation. They leads to the improvement in the focus, in the decision making, and self-awareness, and reduce mind wandering, because DMN is also related to the mind wandering. So this all improvement in this function, the better connectivity has been observed, and that leads to this improvement in these areas. Amygdala has been observed is a part of the limbic system into the brain, and it also shown that it reduced amygdala volume, so reduced reactivity to the stress is seen into the meditating persons. The lowered stress response is there. The improved emotional regulation and decrease into the anxiety and depression. This has been observed into the different studies there. We have already gone through all this. In pain meditation, pain is also one of the area, and unfortunately Dr. Vinod Tiwari is working into the pain, mostly into the pain management as well. So this can be a non-pharmacological way of just looking and changing the perception of the pain in there. And it is observed that this meditation modulates the activity of the brain regions involved in the pain processing, like anterior cingulate gyrus, like insular, thalamus, and prefrontal cortex, which are different pathways into the pain pathway in the body, the parts of the brain. So these all evident, and most importantly, the enhanced BDNF, brain-derived neurotrophic factor, which are very important for development of the brain, for regenerative ability of the brain, the neuroplasticity. So this is also found to be enhanced with the meditation and activity. These are conduct activities. These all we have already discussed. I talk one of the meditation, but few slides about this there. On Preksha meditation, we have been talking developed by a combination of the concentrative meditation and all the other type of the meditation I talked about. This combination of these one, 2-3 types of meditation, they are combined together, developed by Acharya Mahaprabhu in 1976. We have done certain studies we have conducted on this one. It was this Tayosarg Antar Yatra and Shwans Pleksha, Sharir Pleksha. These 4 things we have done into the patients in our suffering of the moderate, mild to moderate kind of depression and into the anxiety. So, no time will not discuss. There are certain kind of studies which also study that it also improves the personality, different personality, strength of the ego strength, super ego strength, and the self concept. All they are improved with the person going Preksha meditation. And there are several studies. I will skip into these studies. What is found the application in neuropsychiatry, it has shown that it has shown benefits into the depression, depressive states, into anxiety. In obsessive compulsive disorder, OCD is extremely common. Then mixed anxiety depressive disorder, we have done study. I give 1-2 slides related with the mixed anxiety, because many times we are not able to differentiate with their sub-syndromal type of the anxiety and depression patients combined together. We see into the clinical practice where you cannot differentiate patients suffering from depression and anxiety. Both the features are there, but not to that level where we can diagnose the as a new clinical entity that's suffering from depression and anxiety. So that is also very common. These are the components of this meditation. One of the study we conducted here, one of the PhD scholar, and we use the Preksha meditation for 3 months. With the follow up on every 1 month, and there was a notable decline to the depression, anxiety, and stress level was seen as measured by DAS scale, and there was 60% reduction in the emotional distress parameters was observed in this condition, and these findings are again elaborated on this one. On 120 patients we did, and the 40 patients they are divided to the 40 patients randomly into 3 groups. And there one group we give the Ayurvedic Medhyapati, and other we give the lifestyle modification plus the Medhyapati, and the Preksha meditation group also we give the Preksha meditation. But in this group, both all the 3 groups, there was a notable decrease into the anxiety, depression, and stress level as measured by DAS 42 scale, and there was maximum reduction was found into the this Preksha meditation group. And this study was conducted into the stress syndrome, and another study this we conducted on mixed anxiety depressive disorder, where again we find that there was 62% reduction to the depression and similar level into the stress model, and we also found that apart from this improvement in this one, we found that the WHO quality of life scale also, we found the physical, physiological, social and environmental well-being was observed when people practicing Preksha meditation for 3 months, this kind of meditation. So this is extremely important. This is practice for. Then the 66% patients in the Preksha meditation group, they showed complete remission of the dysphoric mood. This is most main component of this depression, which is where the sadness of the mood is extremely observed. And also we found that there is improvement in the WHO quality of life. Here quality of life improves, which in all the major dimensions, we include the physical, psychological, social, and environmental dimensions we find. That is certain data and certain group, and there is percentage reduction to the into the depression, anxiety, and stress level in the patients of the mixed anxiety depressive disorder, MDD. So enhanced quality of life is one of the important findings which find into the PTSD, in the chronic pain and the mood disorder has been found, and meditation has been associated with the enhanced well-being, improved sleep quality, and greater resilience. And even the meta-analysis study comprising of these several randomized clinical conditions have shown that the mindfulness meditation has got moderate evidence of the improvement in anxiety, depression, as well as in the pain perception of the pain is there. So and the cognitive behavior therapy is almost similar to response to we find with the cognitive behavior therapy, and the strongest evidence is for improvement to the attention and the executive functions and the skills we find there. These are the overall uses of meditation we have already discussed. Lastly, we can conclude that not only the physiological level or functional level, structurally also the meditation has been proved to increase the cortical thickness and gray matter density, notably into the prefrontal cortex and hippocampus and insula, which are responsible for the attention, learning, and emotional regulation. It's important. And then in the functionality, the default the increased connectivity has been observed into the DMN as we observed and activity control circuits. So this will lead to the reduced mind wandering and improve focus on our better decision making. And similarly, it modulates the amygdala, which reduces its volume and its reactivity, that lowers the stress level, anxiety and depression. Again, the meditation boosts the neuroplasticity by elevating the brain-derived BDNF level and uniquely engages the pain modulation circuits to reduce the pain functions. So, and it has got the cognitive benefits, it has got the psychological benefits in the different kind of clinical conditions, and all the clinical applications it has got in the different mental ill health, in the chronic pain and stress resilience. We can get the very effect. With this, I thank you organizers, especially Professor M.K. Thakur sir, Dr. Vinod Tiwari, Dr. Rajneesh ji, and all those organizers who have been there. So thank you all of you for participating. This was the non-pharmacological approach can also bring certain kind of dividends to us, like meditation. The similar kind of results have been found. Just we get from the herbal medications or the chemical medicine. Certain changes can be there because the mind is our most powerful thing, and whatever our thoughts are there, they are directing our activities. In Ayurveda says that the Saktam Uchate Mana Sariyas Tantrakam Atmasya Yoga. That is also all has been defined in all these activities. That mind is the most powerful thing which controls all the systems of the body, and with this changes in thought process, changes in our perception, all this we can change, we can bring changes into the different kind of functions. So with this I thank the organizers there for me.
Thank you so much, sir, for your insightful talk on the meditation as a neurotherapeutic tool. Now I would like to invite Dr. Anurag Vasne, sir, to facilitate Dr. J.S. Tripathi, sir. Now we will proceed towards press talks by young investigators. The time format will be 15 minutes per talk and an additional 5 minutes for discussion. The speakers are requested to kindly adhere to the time limits. Our first press talk by Dr. Ashutosh Kumar Dubey. Dr. Ashutosh Kumar Dubey is an associate professor and the current head of the department in the Department of Ceramic Engineering at IIT BHU Varanasi. His research expertise lies in bioelectronics, piezoelectric bioceramics and nanomaterials and functionally graded biomaterials, particularly for bone and orthopedic applications. He has led groundbreaking work on piezoelectric composites, exploring their electromechanical and antibacterial properties, electrical stimulation effects, and cytocompatibility for tissue engineering. He is also the author of a CRC press monograph titled Interdisciplinary Engineering Science, Concepts and Applications through Materials and Material Science. Beyond research, Dr. Dubey supervises graduate and postgraduate students, contributes to interdisciplinary teaching, and plays a key leadership role in the department administration and national research collaboration. Let's welcome him with a huge round of applause.
Is there any break? Actually, I have been told that after this, anyways, shall I proceed?
Tea will be served here only because we are running late actually, so we'll try to
okay, thank you.
by 6. Yeah.
Okay. Okay, so I will adhere with the timing and I will move little bit faster. So if you have any question or concern, please contact me later on. So if you see the human body, so almost no process in the human body takes place without electricity. Even when we are resting, you can see heartbeat and prices of oxygen into lungs, and activities of brain, they all takes place with the help of electricity. And these phenomena in animal and plants are known as bioelectricity. As you all know that, this, okay. So, this ionic gradient across the membrane is mainly responsible for bioelectricity. So if you can see this in the middle, so this is the Maxwell's third and fourth law of electrodynamics. So you can say that these law actually tells that if the electric field is changing with time, it creates magnetic field. And if the magnetic field is changing with time, it creates electric field. That is okay. So, and we know that we have ions present inside the cell and outside the cell, however with different concentration and mobility, right? So that's why there is a transmembrane potential, and that's why the electric field inside any kind of cell, and consequently the tissues and organs. And because they are moving and they are with some cases they are being accelerated. So that's why there is electric and magnetic field both. So you can see the table in the left hand side. This summarizes the tissue conductivity and permittivity value. You can see that nerve also contains a significant value. In fact, it is one of the highest electrically stimulated or excitable cells or tissues. You can see the right hand side of the slide. This summarizes basically the range of biomagnetic field, which suggests that human heart is the strongest biomagnetic source in the body, which is followed by the muscle, eye and brain. So here you can see that we are an electrical entity. If we are an electrical entity, that means we can be coupled with an external electric field, right, to stimulate the functionality, to treat the diseases or to correct any disorder. So how to proceed? So initially we started with a cell. So because we are an electrical entity, so initially we designed the electrical equivalent circuit of a cell. So how this electrical equivalent circuit of a cell was designed. So it was designed based on the path of ion traversed under an applied potential difference. So under an applied potential difference, here actually we have extracellular matrix. So extracellular matrix because there are ions, protein molecules. So these lipid molecules, they usually hinder the movement of these ions. So they can be represented as capacitor. So here the extracellular matrix was replaced by the parallel RC network. And this cytoplasm and nucleoplasm are by resistors. And cell membrane and nuclear membrane, because they create the hindrance in the passage of ions, so they are replaced by the capacitors. So I am not going that much into detail, but like this the electrical equivalent circuit was designed. And by considering another pathways, ionic pathways, we have designed many other electrical equivalent circuits. And if there is any model, it requires certain assumptions to solve this model. So here there are certain set of assumptions. Again, I am not going to these assumptions step by step. And then we solve actually this circuit in order to get the time constant of this particular equivalent circuit. Right? The time constant has been calculated because we want to understand that how this electric field, how this cell can interact with the electric field. I mean, if you want to take the advantage of external electric field, so we have to calculate the time constant of this cell. So we got the values in terms of, various resistances and capacitances of cytoplasm, nucleoplasm. Then we took certain assumptions, and based on this, we got the numerical values of various component of, cytoplasm resistance, nucleoplasm resistance, cell membrane and nuclear membrane capacitances. And then finally we got the time constant for the cell, that is 2 to 5 microsecond. That means the cells are very prompt towards the externally applied electric field. And in this model, actually, we considered the cell membrane and nuclear membrane as capacitors. But actually, they are not the capacitors because, they allow the passage of certain type of ions. So the sodium ion, potassium ion, calcium ion, especially these, excitable cells like neurons, they usually allow. So in order to simulate that, actually, we considered them. We modified our models, and we introduced them as IITs. So then we finally calculated the time constant, and it was around 3 to 5 microsecond. So it is of the order of microsecond in any case. So we have modeled in many ways the electrical equivalent circuit of the cell, but in all the cases we got this value of the order of microsecond duration. That means the cells are very prompt towards the externally applied electric field. And then we try to understand what is the implication of these models, because the cell can, during its processing, the cell can decrease or increase its cell size. So how this time constant will vary when the cell size is changing. And then we calculated actually how the time constant varies with these parameters. So you can see here the nucleoplasmic resistance, they show more stiff behavior as compared to cytoplasmic resistance. So again, I'm not going that much into detail. And here you can see the time constant variation with the capacitance of cell and nuclear membrane capacitances. You can clearly see that the nuclear membrane capacitance shows more stiff behavior as compared to cell membrane capacitance. Then we try to understand what will happen if we are going to apply this electric field on cells. So and we know that if we apply the alternating electric field, it will have a lot of heating effects. So alternating electric field has no advantage. Actually, it has disadvantage. So we chose pulsed electric field. So we choose millisecond duration pulses, microsecond duration pulses, nanosecond duration pulses. So those results are not here. We got the best result with microsecond duration pulses, and we we applied 1 volt electric field, and you can see in all the models, most of the actually applied voltage or current, consequently, you can see they are across the cell membrane, and it is not affecting the nuclear membrane. So this is what actually we wanted if you want to stimulate the cell functionality, or if you want to treat any kind of disease or disorder. So this is just another computational study that we got during our modeling study. This is very interesting result, as we know that the normal and cancer cell have different electrophysical parameters. So that means they require different electric field to electroporate. And if you already know what is reversible and irreversible electroporation. So we calculated this for irreversible electroporation, where you can kill the materials, like any kind of cells. So here you can see that for a particular combination of electric field intensity and pulse duration, you can basically kill the cancer cells selectively while keeping the normal cell intact. So this is the beauty of electric field treatment, is that you can keep the normal cell intact and you can kill the cancer cell selectively. So, and because we have developed model, these are all theoretical models, so we have taken because there are number of studies available on electroporation. So we have taken actually these experimental results from number of studies, and we have validated our models. Now, based on our modeling studies, now we have all the parameters from cell. We used all the parameters to design the electrical field stimulation setup inside the lab. So these are the setups that we have developed. So we are using, mostly people use circular plates, right, for the cell culture. But because we are applying the electrical field, so we need rectangular. So we are using this rectangular tissue culture plates. And there are two ways by which we are stimulating the cells. One is you can apply the electric field. You can apply the electric field during the cell culture, and another one, if your material is piezoelectric, you can basically charge the material before cell culture experiments. So this is the sequence of electric field. Actually, we allow the cells to adhere, and then we apply the pulsed electric field just for 5 minutes. Again, we allow the cells to adhere on the incubate it for some time, 12 hours, and it depends on cell type. And again, we apply the electrical stimulation, and mostly we apply the electric field parallel to the sample surface, perpendicular. We tried perpendicular also, but the best result we got when we are applying parallel to the sample surface. So as you know, the piezoelectric materials, they can be charged. So what we are doing, we are taking the advantage of charged surfaces as well as the application of pulsed electric field. So we are allowing the cells to adhere on the negatively charged surface, and then apply pulsed electric field. We are allowing the cells to adhere on the positively charged surfaces, and then applying the pulsed electric field. And the best result we got, when the cells are allowed to adhere on the negatively charged surface and electric field is applied parallel to the sample surface, not perpendicular. So this is one of the ways, but in this way actually we are not getting good result. We are getting the best result when you apply the electric field parallel to the sample surface and cells are being adhere on the negatively charged surfaces. So these are just the result actually on materials perspective. So I am skipping this. And this one of the very good result that we got. If your surfaces are charged, they will, they accelerate the initial sterilization process. So we apply this, the combined action of electrostatic and dynamic electric field on mesenchymal stem cells, and we got, as I said earlier, we got the best result when we allow the cells to adhere on the negatively charged surface and pulsed electric field is adhered while the cells are being adhered. This is one of the study that we did using, this PVDF polymer. As we know, the PVDF is a piezoelectric polymer, and actually the beta phase, the beta phase of PVDF is piezoelectric. And sodium potassium niobate, this is the piezoelectric ceramics that we developed in the lab. And what we did, we developed this fibrous sample using electrospinning, and we took embryonic mice at embryonic days, 16, 17, and then we took this hippocampal neurons and we performed this study on the charge when these fibers are charged using the hippocampal neurons, and we got very good results. That means your dendrites and axons, they follow a very beautiful path of the charged surfaces. So with this, I would like to thank the organizers for inviting me. Thank you very much.
Now the session is open for discussion.
When you culture the cells like those which are not that much known to be polarized in respect to neurons, was there any difference, or do you culture neurons at all?
No. Actually, neurons are the most sensitive cells against any kind of electrical stimulation. So we used osteoblasts, we used fibroblasts, we used neurons. So osteoblasts, actually they are they're least responsive to external electric field, so they need very high electric field. And neurons, osteoblast 1 volt per centimeter you can excite it. Osteoblast, neurons 0.3 to 0.5 volt you can excite it.
You cultured the neurons where they faster with proliferation or something.
Yeah. So almost this doubling time has been reduced by 50%.
What about the cancer cells you talked about? They have a piling up feature. So
Cancer cells we have not done actually. Experiment that was theoretical study. So yeah.
Okay, like we have used them as a whole cell, that in the biological study, where we are taking is like the circuit model use, the component wise. Where we are considering somewhere to be like capacitor, there is a resistance. So are there some models based on that?
Models means a lot of people they followed our study. Actually, yeah, and we used our study to develop this electric field setup to stimulate the cells, and we got very good result. And using our models, we have calculated the strength of electric field to required to electroporate the cells, and we compared all these electroporation studies that is available experiment.
Your model was based on the cell type, and you designed it like the circuit unit.
Yeah.
So therefore, electroporation meaning for doing those micro manipulations in cells. I don't know whether that electroporation you are using now for that.
Actually, we have there are certain parameters for electroporation, right? So using our models, we calculated the strength of electric field required to electroporate the cells.
So like in our field, we do lot of like plasmid going inside the cell. So will it help it for that?
This electric field strength, yeah,
Will help.
yeah.
So first of all, it's like very nice, very clear. I have like one query and one suggestion. Query is that when you are calculating this electrical field parameters, you have taken sodium, potassium, calcium as drivers. But all three of those cations have a differential permeability. Right? So that will mean the symbolic battery that you have put it on would have a differential value as well. Have you encountered that, or you have all together?
Oh, we kept them all together because this study actually this calculation was around 80 pages. So otherwise the calculation, if you are going more deeper, your calculation will become more complex. This is actually third order differential equation. It's very difficult to solve. So if you can consider more, actually it will become more complex.
So when you go for the null equation, which comes with the capability of looking at permeability at the same time, concentration gradient, you get some more sizes.
Yeah, that gradient we have.
Good. And second is the comment, because you see fantastic to see the differential size growth. My thought was that if you put cardiomyocytes in these plates, you have a thing, then you mention heat. Because we need electrical stimulation to be in. So there is one piece we understand that would be the whole heart. So if you can use these
yeah, probably, but we have not seen it. Maybe we will try it, yeah.
So there are some sort of SNCI, they do very well the culture, and some of them start to be. So if you put them in this system, it will be better, we will have this one side. Thank you. Very good.
So like when you are using the signal today, they are already positive charge for the system at an idea and nothing more so particular. So you are doing some simplification, so you can use
Negatively charged, yeah. What we are using the piezoelectric substrates, and we are bombarding very high energy electrons to make it charge. Yeah, to make it negative. We are inducing the charge.
for the sensors also, like they want to deliver on that, like after multiple layer of code, they want that to be also.
Yeah, but for that actually we did theoretical studies, experimental studies here. Okay, thank you so much.
Thank you, sir, for your insightful talk on piezo biomaterials and their electrophysiology for cancer treatment and peripheral nerve degeneration. Now I would like to invite Dr. Amresh Singh, sir, to felicitate Dr. Ashutosh Kumar. Yes, sir. It's my pleasure to introduce Dr. Amresh Kumar Singh, sir, Associate Professor at the Department of Zoology at the Banaras Hindu University, Varanasi. Dr. Singh is a leading researcher in the areas of osteoarthritis, inflammation biology, and the hormonal regulation at the bone health. With a strong background in the vertebrate physiology and experimental pathology, he heads a research group dedicated to understanding how bones degenerates and especially in osteoarthritis. At BHU, Dr. Singh is also deeply involved in mentoring young researchers, developing academic programs, and promoting interdisciplinary collaborations. His contributions and helping bridges the gap between laboratory research and real-world treatment for musculoskeletal disorders. Please join me in welcoming Dr. Amresh Singh sir with a warm round of applause.
So we also have to conduct a poster session or maximum about that. So we will take that because of shortage of time. So poster number from 1 to 8. So participants on both coalition can go to their posters. Eventually will be coming to your posters and be talking to you, discussing. Okay. And then after 1 to 8 is done, then we'll go for the another 6 posters as well. Okay.
So very good afternoon. Yeah, I'm Amresh Singh, and today I'm the odd man out who is going to talk about a different aspect of disease which is not at all related with your brain. But I must say, if your joints are not working well, your functional brain cannot help you out. In that condition, I'm going to talk about a disease under two phenomenological conditions where the BMP signaling and macrophage inflammation is taking part. Before going to that, I would like to say there is a word showing here that is pain, and I am very much confident nobody will like this word. So the question is, do this public like? I guess no. The disease leads to a life with disability, and this also causes a huge economical burden to the society. Not only that, in 2016, the OSRI society has designated this disease under serious diseases condition, and they have given some characteristic, and they mention there is no cure till date. This causes huge disability, loss of productivity, huge loss. There is very least interventions are available which are very costly, and the associated side effects are huge. Ultimately, they have given a definition that osteoarthritis is nothing but it's an inflammation of joint, where if you see this is a normal joint looks like, and the tissue which get affected here, that is known as articular cartilage. And as the disease progresses, this particular tissue get damaged, and ultimately your joint gets damaged. There is only one option, that changing the joint. The structure what you see here, that is known as osteophytes, and the tissue I'm going to talk about, that is the articular cartilage. And ultimately, this gets damaged once the disease progresses. Not only that, this society has defined this disease, and they have said that there are two physiological phenomena that is being involved here. One is endochondral ossification, through which your bone gets forms, and second is inflammation that is being involved. Since 3 decades, people are working worldwide, but most striking part is there is no disease-modifying therapy available. I would request you not to record this unpublished data I have, please. And ultimately, what is happening here? There is no disease-modifying therapy available. So there are two schools of thought. One school of thought says there is a repetitive loading that leads to inflammatory mediator appearing in the picture that causes the activation of endochondral ossification mediators, and that ultimately leads to osteophyte formation. Second school of thought in which my lab is working, that says if there is any mechanical insert, that will lead to ectopic activation of BMP signaling, and that causes activation of articular cartilage fade into transient cartilage that ultimately forms bone, that is osteophyte formation. Means what? In osteoarthritis, there are two biological phenomena being involved. One is inflammation, another is endochondral ossification. Question arises whether that inflammation precedes endochondral ossification or endochondral ossification precedes inflammation.
That is a $1 million question still unanswered. And most striking part, both group of scientists says, believe me, we are saying the truth. But what is their condition? There is no cure. Ultimately, how I study the disease when this is occurring in old age or in a post-traumatic condition. For that, if there will be any abnormal loading, you will see this picture. If there will be any abnormal loading, that will leads to abnormal mechanical load on your knees. And ultimately what will happen? It will trigger the damage of articular cartilage. And ultimately, how I am mimicking this? I perform a microsurgery in the joint of mice, that is survival surgery we do. And ultimately what we do, we cut the ACL and try to mimic the disease. To check that first, what we have checked first is what will happen if you just surgically induce osteoarthritis. To the factor which what I have considered, that is endochondral ossification mediator, BMP signaling, as well as inflammatory mediators. What data we have found is that once we have induced the disease, you can read my data as post-surgical day 14, post-surgical day 28, means I do temporal study, time-dependent study. And what we have noted, as we have induced the osteoarthritis, BMP mediator, that is a readout of BMP, phospho-SPAN expression started appearing on day 14 and 28. Then we have checked the characteristic marker of articular cartilage, that is collagen type 2, and we have noted as the disease is progressing, the cartilage, the collagen type 2 level is going significantly down. Ultimately, we have checked the matrix mediator as well as the hypertrophy mediator, and we have noted as the disease is progressing, these molecular markers are appearing in the picture. Ultimately, we have checked another component, that is the inflammatory mediator, and we have seen that in terms of NF-kappa, IL beta and TNF-alpha, we have noted as we have performed the surgery, they are appearing before early compared to day 14. They are appearing in day 7 itself. So it means our model is mimicking what we want. Coming to the next condition, I said there are 2 players. In that condition, what if I can remove one player and see what is the other player doing in the game? In that condition, we have used clotronate liposome that actually macrophages they engulf that liposomes and they get died. And we have confirmed this expression by doing a clotronate liposome injection in the intra-articular space. What we have noted here, this is a control condition, and when we have treated, you can see 0-1 data that all the synovial macrophage lining got disappeared in terms of F480 expression. It means one player is not at all in the picture. Coming to the next condition, what we have seen, now we started checking what is happening to the endochondral ossification markers. So first we have again reconfirmed what is happening to the macrophages. Then we have checked the expression of hypertrophy marker, COL10 as well as collagen type 2. And what we noted here, as the disease is progressing at each time point, once we are injecting the clotronate, there the hypertrophic marker is going significantly down, and even the COL2, which is a characteristic feature of your articular cartilage, is improving. Ultimately, we have checked the inflammatory mediator as well, because we don't know whether only the macrophages are coming in the picture or is there any infiltration as well. In that condition, we have checked at each time point, just like day 7, 14 and 28, the expression of inflammatory mediators, TNF-alpha is going significantly down. Then we have checked the INOS expression as well. There could be a nitrosative load. That is also significantly down. Then we have checked what is happening to the macrophages that may appear in the picture, and their level is also going significantly down. And NF-kappa being a central mediator of inflammation, their level is also significantly going down. That is not our target, that people are saying. Ultimately, what we have done, we have checked for the histomorphometric analysis, what is happening at the B site. Then, what we have seen here, at all the time point, not to even that, at the longer time point also, their osteophyte formation has significantly gone down. If you see this picture and if you compare to this, your joints are comparatively better as the disease condition is. We have reconfirmed this data by doing the toluene blue staining that says the quality of our articular cartilage, and that got improved. Ultimately, now the picture is we have removed one player and seen what is happening. In another condition, what if, because the previous one I have induced the model by doing the surgical induction. Here I have utilized the system where there is no surgical intervention. Directly using the transgenic mice, I have induced the spatiotemporal BMP signaling. That is a non-invasive way. What we have done, we have a mice where we can directly induce the BMP signaling at the target site. That is a collagen 2. Wherever there will be collagen 2 expression, there I can induce the BMP signal. So what we have done here, we have removed one factor before induction, that is we have given intra-articular clotronate liposome injection. So macrophages are gone from the picture. Now we have induced the BMP signal. What we have observed here, first we have checked whether the BMP signaling has been induced or not. For that we have checked the phosphospan 158 expression and seen that even day 7 signaling started appearing in the picture. As the day is progressing, the disease is progressing without any traumatic injury, without any surgical induction. Coming to the next condition, we have checked what is happening to now collagen type 2 as well as the hypertrophic marker. We have seen at each time point the call to expression has gone improved. You can comment here because at day 56 improvement is not that good. I would like to tell you it's a transgenic mice, induction is genetic, but still there is an improvement. Removing clotronate, removing the inflammatory mediators is improving the condition. Ultimately, we have checked what is happening to the hypertrophic marker in terms of Colton expression that has gone down at each and every time point. Coming to the next, we have checked the metalloproteases expression. Their expression is continuously increasing as the day is progressing. We have checked up to 6 months. Then we have checked the expression of the inflammatory mediators, that is the TNF alpha in all the time points, and they are keep on increasing as the disease is progressing. Means what we have done, we have removed only one factor from the picture. Then we have performed the histomorphometric analysis. Here if you see, this is a condition that is a post-induction day, means no surgical intervention. So disease is progressing as the day is passing on. So up to 6 months we have checked. Then we have given a clotronate liposomes at all the time points. What we have seen that is very surprising to us, there is improvement. There is a significant improvement if you see that osteophyte formation has significantly gone down. I will not say that it has treated the disease, but it has halted the progression. So halting the disease also is a success because till date nobody can halt the disease as well. They can provide you only palliative care and the diseases keep on progressing, your joint fails, then your brain will not work. This data we have further checked while using the fundamental staining, that is safranino and toluene blue, that actually says about the quality of your articular cartilage. That has also been improved once we have removed the factor. So what I can conclude here, that scavenging one factor from the picture is declining the kinetics, and both are participating here, equally participating. And what till date clinicians are doing, they are targeting one at a time. So that's the one reason there is no success. The work is still in progress. We'll give you more update in coming days. So what could we take home message? If your knees are weak, then what you should do? Don't start a sudden exercise because it's a mechanical tissue and that will kill your knees. Why I'm saying? Because each and every activity of yours has a different load on your knees. So ultimately sudden exercise will kill your knees. Don't do that because you will induce mechanical insult immediately. And if your condition is belonging to these three categories, here in a healthy condition, the inflammation is low, systemic inflammation is low. But if your condition is this, your mechanical stress is very high, muscle strength if you see, that is low, and ultimately what is happening here, one factor is available. That is a systemic inflammation. So this will provide a situation like Abhimanyu will be compromised. Fine. Coming to the next, what shall we do? Do the muscle strengthening exercise. If you do the muscle strengthening exercise, what it will do? It will actually hold your joints. That will provide a stability, so no abnormal loading. So ultimately what you are doing, you are saving your knees. And these are the heroes due to which I am standing here. Thank you very much.
Now the session is open for discussion.
Sir, I might not have followed you, but
Yeah,
as
please.
usual, a very nice pictorial presentation. So that creative factor expression आपने जो पहले दिखाया, is it going down in 28 days?
It is going, no. In the first step, what I have shown, I have done a mechanical insert. Then I have just checked what is happening, whether it is following the pattern or not, whatever the data says, whatever the literature is saying, right? In the second condition, I have removed one factor. Removing one factor will not provide you assurance that your whole joint environment is safe. There are other players also. So that's the only reason we have checked all the time inflammatory material.
तो ये creative factor नीचे क्या?
Yes, if you just give clotonate liposome, definitely it will go down. Yeah, please.
It was like very good data, and in real terms, I was like very well done. My query was that in addition to looking at the bones, have you also looked for the synovial membrane, for the hyperplasia frequency?
Synovial inflammation we are talking about. Synovitis. Yes.
How the membrane structure was?
It changes. Drastically changes. If there will be any synovitis, in general, normally the membrane is single lining or double lining. That's it. Okay. If there is any kind of inflammation, the lining goes thicker and thicker.
Have you seen?
Yes, we have seen. We have some more other data also. But what here I'm talking about the two factors which cross-talking with each other. So that's why I've not shown the data.
You might achieve those numbers.
Thank you so much. If no more questions, thank you so much.
Thank you, sir, for enlightening talk and clearable vision regarding the inflammatory disorders. Your thoughtful insights have created great depth in our understanding towards the pathobiology of joint diseases. Now, there is a next flash talk by Dr. Gaurav Kumar Pandey. Now I would like to invite Dr. Gaurav Kumar Pandey sir to felicitate Dr. Amrit Kumar.
It will be a good frame actually. Thank you so much.
Now it's time for our next talk by Dr. Gaurav Kumar Pandey sir. Dr. Gaurav Kumar Pandey serves as an assistant professor in the Department of Geology at Banaras Hindu University, Varanasi. His research expertise centers on chromatin modifying enzymes in cancer progression, long non-coding RNAs, epigenetics, functional genetics, and drug development. With a solid academic foundation, including a PhD in geology from Banaras Hindu University, Dr. Pandey investigates molecular mechanisms underpinning tumor biology and gene regulation, often bridging foundational epigenetics insights with translational potential in therapeutics. He has authored numerous peer-reviewed articles and actively mentors undergraduate and the graduate students in advanced molecular and genetics research at Banaras Hindu University. Let's welcome him with a huge round of applause.
Thank you. First of all, I want to thank all the organizers, Dr. Vinod, Dr. Rajneesh, for giving me this opportunity to share my work. We are, as a group, very interested in understanding epigenetics, and we take any disease model. So we work on cancer as well as infertility, but the core area of work always remains epigenetics. As this is a talk on brain and neuro disorders, I will share with you a data which was done earlier by me in a disease or a cancer related to brain. So what I am going to talk today is about a cancer which is called as glioblastoma multiforme, or in a layman's language, this is called as brain cancer. Now, amongst all the deadly cancers, brain cancer is very aggressive, and despite having classical therapies like surgery, radiotherapy, or even chemotherapy, the outcome is extremely poor. If a patient has this cancer, and we'll also show you by a mouse model, the chances of survival is barely 10 to 11 months, and despite giving any kind of treatment, the most that patient will survive will be another 3 to 4 months. The problem with this cancer, or what makes life very tricky from a researcher's perspective, from a clinician, is that it is a highly heterogeneous tumor. So classically, like many other diseases, this was divided as a low grade, mid grade, or a high grade tumor. But now, from a genetics perspective, what we see is that we can classify this into 4 subtypes. And when we say subtypes, in today's day and world, we use exact molecular signatures, where nowadays we can divide a glioma or a glioblastoma into 4 exact subtypes. One is proneural, which has these classical markers like loss of P53, which is a known tumor suppressor, IDH1 again loss, and ETGFR. Then we have mesenchymal, where you have loss of NF1 and P53. Classical type is called where you have activating mutation in EGFR, another oncogenic mutation, and CDKN2A. And finally you have neuronal genes. So what you can see is that it already signifies that there are 4 parallel diseases running. And one of them can be there, or sometimes you can also see phenotype of 2 of these. Now, why it is so difficult from a clinician perspective as well as from a researcher's perspective to work on glioblastomas? The first, as I have explained, it is an extremely heterogeneous, resistant kind of disease. You will see heterogeneity at genetic level, expression, multiple levels. Second is, whenever you do any kind of research, it can be preclinical. If you do research in animals, you can make multiple discoveries, but the problem is, if your target molecule or your discovery is unable to pass through blood-brain barrier, it has no meaning. So many times what happens is, we see in publication, you get very happy, which we should be, that your inhibitor is working on the target extremely first class, but it cannot cross blood-brain barrier. Third is lack of good model system. What is a model system in which we are going to study this? Majority of time you will see that people use cell lines thinking that this is glioblastoma, but they grow them in serum. If you are growing your cell line in serum, it is not glioblastoma. The moment you grow it, it will differentiate, and the litmus test for anything which you claim as a model of glioblastoma is you make an injection in animal, and if it is unable to make tumor, then it is not there. Finally, another very important thing is, this is an expensive system, because we grow glioma stem cells, the media reagents are extremely expensive. You need a very good skill set to make experiments in animals, and a lot of other strategies. Now, what we have tried to do is, as I initially mentioned, is that my interest is epigenetics. Now, with the possibilities which have been already explored and they have failed, epigenetics comes into rescue, because if you see this picture, this kind of summarizes what happens in this cancer. You have metabolic abnormalities, you have genetic abnormalities, which I have explained, and then you have tumor microenvironment, you have immune players, CAF, and so many other things, interleukins. All of them are interconnected, and they influence a common goal or a common mediator, which is the epigenetic molecules. For students, when I say epigenetic again and again, you primarily mean 2 things. One is modifications on histone tails, or addition or loss of DNA methylation on gene promoters. So if you see this cartoon, what naturally should happen is that you have neural stem cells, and they have a particular chromatin profile. They would slowly divide and go into these different type of differentiated cells. But what happens is, in case of gliomas, they will become brain tumor stem cells. So when in lab also we grow them as stem cells, but they are now different than your stem cell, and they have the ability to cause cancer, and they have got this cancer stem cell like hierarchy. Why we are interested so much into epigenetics is that 46, almost half of the patients who are affected by gliomas have mutation in at least one of these epigenetic regulators. And the drugs which are available against these epigenetic molecules are very well tolerated, so they show less toxicity. Then the model. What we started is, we took our model from a published resource, and this resource is publicly available, so they do not sell it. It's not a company's property, it's a university. You can write to them, and if you convince them that you are a researcher, they can send you for free. What they have done is, they have developed this human glioblastoma cell culture resource, where they have isolated cell lines from patients, characterized them, and again when they inject into animal, the injection or the tumor which appears is quite similar to what was in patient, so it matches the profile. It is able to make tumors into when transplanted into mouse brain. So then these human glioma cells are one of the good systems to explore or make discoveries. So we have now gone through the problem and the model system that we have used. Then what exactly we did? So with a background in genetics, what we took refuge into is that we performed CRISPR screens. And we took two kind of libraries. One is libraries targeting kinases, against which many drugs are available, and a library targeting epigenetic genes. So for students, what or how do we do a screen? I will just explain it briefly. What we do is we take cells and then we transduce the library into these cells, and the idea is that you make this transduction in such a way that one cell will receive one guide RNA. So if one cell receives one guide RNA, what happens is then you grow these cells. At end of your experiment, there will be certain cells which will lose these guide RNAs. And if they lose these guide RNAs, by that we indirectly mean that the cell is unable to sustain the loss of that gene, which again indirectly means that if you target those genes, they can be a therapeutic target. How do we do that is, every guide RNA is labeled with a barcode. So what you will do is, in the beginning of your experiment as well as at the end, you will amplify these guide RNAs, go for next generation sequencing, and then use MAGIC, one of the softwares or any other software which you are comfortable with, and then look for what are these depleted candidates. What we did is, we took a subtype of glioblastoma, two cell lines we purposefully took, proneural 1 and 2 is labeled here for understanding. We transduced them initially with a KRAB-D-Cas9, which is Cas9 is needed for any CRISPR to work, and this Cas9 is tagged with M-cherry. So you can always look under microscope whether your cells are carrying Cas9 or not. Second advantage with this system is, this is DOX inducible. So whenever you add doxycycline, you can switch on the system and then you can switch off if you wash away that media. We make these transductions and then we grow them for 3 to 4 weeks. One is DOX uninduced, so it's a control, and then we induce DOX and then look for which genes are depleted. A very brief thing about it is 500x represented. Every library has its specific genes, and also every library has two controls, guide RNAs against essential genes and non-essential genes, to look for your readout at the end. This is our outcome. Initially, when we analyze our data from proneural cell lines from epigenome screen, you can see that at the end you have these dots which are below. These represents which are the guide RNAs which have dropped out, meaning these are the knockouts or the loss of genes which the cell is unable to sustain, and at the top you have genes which are enriched at the end. Secondly, same thing we did in another cell line, and then when we compare, so this is data of two independent screens, we look for commonality and we get this set of 38 genes where we get many interesting hits. You can see this by network, CAT5, TAF1, many of them. But then again we came back to that question which we started with this, even if we get multiple hits, can we target them pharmacologically or not? And problem with epigenome or epigenetic drugs is very few of them are available. So we have chosen a couple of them only, which I'll talk later. Parallelly we did these screens in kinome as well, and doing this in 2 cell lines, we again got multiple hits. Yeah. So when we consolidate our data from epigenome and kinome, and then we also compare it with a genome-wide CRISPR screen, what we got is total of 38 genes from epigenome screen and 33 from the kinome screen. Many of these genes showed overlap with the earlier published one, which shows that we were in the right direction. Now, everything fine, we have a good model system, we have done in vitro, but then at the end of the day it is still an in vitro system. We are culturing cells in a dish and it has its own limitations, and it can never represent an actual biological system. So then what we have done is that we have tried to recapitulate this in an animal model as well. So what we do is that we inject these cells which are with the library into the making an intracranial injection in the animal brain, where we take a immunocompromised not-skid mice, and you can see this by the picture where we take MRI at 4 weeks and 6 weeks. This is an extremely aggressive cancer. In animal itself, you can see at the 4 weeks itself a small dot starts appearing, which is pretty big in size, looking at the proportionally the size of animal, and by 6 we can see already there is a full blown tumor. So what we did is we injected these cells, wait for almost 10 weeks, and then see what happens. Again perform the same screening strategy, and eventually we got these dropouts and enrichments. So what was the final outcome of all this exercise? What we get is a set of genes which are pretty well known: DNMT1, HDAX, CAT5, PRMT5, TAF1, and these are the hits from kinase screen, AKT, mTOR, PI3K. Many of them you would see that these are pretty obvious ones, and many people have seen. What next step we took is that although these cells have this genetic vulnerability, if you use a single inhibitor, it is never ever going to work. So what we have to come up with is a combination. We have, just in interest of time, we started working on 3 of these genes. One is TAF1, which is a Tata box binding protein associated factor. DNMT1, which is a maintenance methyl transferase, and PI3 kinase is a very well known signaling molecule which promotes cell survival. The reason for this was that the drugs which are available against these 3 target genes. TAF1 has a new drug CMMEC13, which it works at very low concentrations. DNMT1, many drugs have been developed, but the ones what we use is a next generation one, SGI-1027. It's a novel small molecule, works fantastically at a lower concentration, shows extremely low toxicity in animals. And finally PI3 kinase. In literature you will find multiple PI3 kinase inhibitors, but problem with 99% of them is they can never cross blood-brain barrier. So this one is the GNE317, which was able to cross blood-brain barrier. These are the publications which have shown these properties which I have described here. Then next what we did is we took this drug synergy setup, where this is an example of dose matrix where we place each drug in IC50 to lower concentration, and in this matrix we look for at what concentration do they synergize. And what we found is that using this combination index, that if we use CMM13 with GNE317, it shows pretty good synergy, but then again this synergy was not as good as what we wanted. If you see that 0.3 to 0.7 synergy is considered as pretty good, our range was 0.6, 0.3. Same thing appeared in another one. Then what we then finally attempted, and this is, it looks very simplistic graph, it took months of repetition to come up here. What we thought is that actually you have to use a triple combination, and this graph shows you that if you use these inhibitors individually, this is percentage of cell survival. You see that only 20 to 25% of cells are affected. If you use these drugs in combination, and it can be any of these three drugs in any combination, the maximum inhibition is 60%, and this is the lowest possible one. But the moment we use them in triple combination, you see that they work fantastically. So actually what has to be done is that these three drugs, which they synergize very nicely, they have pretty good pharmacokinetics, which is suited for a difficult disease like glioblastoma. If used together would be beneficial, and we have tested this into two different models of gliomas, and both of places we see that individually the drugs barely show any effect, but when used in combination they work very nicely. So this is the conclusion where we arrived, is that using genetic screen as well as pharmacological ways, we come with a triple combination of TAF1, DNMT and PI3K inhibition, which could be extremely effective for treating glioblastomas. But because I've said so many good things, I have to also say what is the shortcoming of this approach. So the moment you start visualizing such things in a patient scenario or even in an animal model, you have to repeat all the experiments from scratch, where you have to starting with maximum tolerability doses. Tolerability, we were able to do that, but then again you have to make those injections, test on the mice. But from the in vitro data, it looks extremely promising and has higher chances to be translated in patient samples, So these are the acknowledgements, and I'll be happy to take any questions.
So I have two
Yeah,
curiosities. Like, first, every individual physiology is different.
Right.
So it means the microenvironment will be entirely different. So how they will act in that condition?
Who will work?
Your drug, so your combination.
See, you have to understand that this cancer, the person is surviving only 1 year if that person is not given any medication, right? If you give medication which is prescribed by doctor, that person will survive another 3 months. So there is a lot of scope of improvement, and when you say microenvironment, we are not touching anything associated with microenvironment here. Microenvironment is one of the properties associated with tumor. What we are touching is the genetic part and the epigenetic part. So both, or like AKT signaling, pretty well established, has a role in glioblastoma. Changed epigenetics has pretty well established, and TAF1 also inhibitors have shown to have pan effects. So in that context, I think it should work very well.
Oh, that is fine. You are saying you are not touching that part. Meaning our ultimate our goal is to treat the patient, right?
No, that is a misconception. What we want is to improve the survival. See, there is a big difference. The moment you say you want to treat is, you would imagine that the tumor is completely gone. That's what I'm trying to explain, that this disease is extremely tricky. And with the current data, I don't think anything would work. All the conferences I have been to, the end conclusion was there is no treatment. So the strategy is just to improve the survival. How much more you can push the system. And the problem is, because it's an extremely metastatic disease, it develops very quickly. The moment something is full blown, I don't think it is possible to eradicate it.
I have that, I want to understand the synergy you said, right?
Yeah.
So how you checking that?
So
like taking two drugs together, what kind of...
So what you do is, suppose you take this wells, one side you start adding from IC50, which is like the concentration where 50% of cells are gone, and you go to the lower concentration, same thing you put the second one, and then in these mixed concentrations you will add your drug, and at some place individually both the drugs might be inhibiting say 5% of the cells, but in combination it could be 40% or 50%. So we use these softwares which can predict that it is a synergy or an additive effect. So if it is an additive, yeah, absolutely. So this is we use 3-4 replicates, because problem is this is done in a 384 well plate. So the chances of variability is extremely high. So you have to make at least four times experiment, and each time it should be in triplicates. And then you analyze, then only you can come. Otherwise these are extremely sensitive and you might misinterpret your data.
Yeah, and this has
Yeah.
contrary to what you told, some of them are highly common ones.
Yes. Yes.
Though I could understand that obviously 8 saal is the limit and you are increasing life by even 4 months is very important. But then how would it affect the normal pathways like your DNMTs and all those are very universal ones?
See,
Except the Aruna kinase and all, they are very
Absolutely.
specific for cancer.
Yeah.
But,
So there, what we can tell is from the animal studies is where we do toxicity, right? The advantage with either DNMT1, which is an epigenetic drug, and TAF1 is they show extremely low toxicity. The PI3 kinase inhibitor, what we are using is at very low concentration. If you see individually, it is barely affecting 5 to 10%. But that will be there. If someone tells that there will be no side effect, is lying. So again, you have to weigh the consequences and cost. So the cost and benefit. If you are getting survival with side effects which are manageable, I think it can be used.
One more thing, just smaller. So you have shown a graph, right, where when the 3 combinations are together, it was very subtle. It was, right, I couldn't remember that graph. So it really affected. So why it's not
Together.
Together, yes.
Yes.
So increasing the lifespan only 3 months.
No, I'm just speculating. We have not done it, and
Okay.
this is cell data. I'm just telling that until unless you do that. So again, if you have, that's what I told, that the drawback is if you have to translate it even in a phase 1 or patients. Nowadays, only a couple of such trials are going where they are using 3 drugs.
Why I'm asking was because last month only 1% died with this particular disease.
I think every day we can say that.
What we are doing at this screening stage?
What?
At the screening stage of these trials,
Yeah.
are we putting multiple targets or one by one the targets we are putting and then combining it for the synergistic?
Sorry, I didn't get the question.
When we are screening the molecules,
We are not screening the molecules. So these hits have come from our genetic screens. So we get these drugs which are suited to this disease. Like they should work at much lower concentrations, they should be less toxic, and they should have the ability to cross blood-brain barrier.
so what I want to do is that you choose one gene, one molecule,
Yeah.
and then the other gene, other
Yeah.
molecule, and then put it in each.
Yeah. But I mean, what I have shown you is one, we tried multiple synergies, like all the hits, and then we came up with the best possible.
Thank you so much, sir, for your lightning talk on the uncovering genetic vulnerability. Prakash Modi, sir, to felicitate Dr. Gaurav Kumar Pandey.
Using GnRH which controls the gonadotropins from the pituitary, and finally the steroids at the gonad level. And this is what the decapeptide which I was talking about being given the Nobel Prize in 1977. It's a very important found in human. Its isoforms are present in fish and chicken, and it's all through evolution. Its isoforms are present in fish and chicken, and it's all through evolution that you get different types of GnRH. It's a decapeptide where these 4 amino acids for designing new novel agonist or antagonist of GnRH molecule. Next question, which is the transcriptional regulation. Being a gene which is alternatively spliced, it can also be regulated at the level of post-transcriptional regulation, and then obviously a protein getting synthesized and then secreted. So these are the levels. This is where it's known that GnRH has a proper sine curve across the life of an individual, because in a young age and an old age it's low, in the reproductively adult phase it's high. So this is well known, we wanted to replicate it, and what we were looking into initially, was just to look into the regulators of GnRH, but then lot of literature is available, so we wanted to venture the non-hormonal regulators or the metabolic regulators, and in that we got interested into two cell types of the brain, particularly the astrocyte and the neuron which secretes GnRH from the hypothalamus, astrocytes which are the partner cells for neuron, giving them the nurturing capacity because they typically nurture the neurons. And there are a lot of crosstalks between neuron and astrocyte. One of the crosstalk that is well known is the shuttling of the metabolic fuel, that is glucose and lactate. So neurons, one of the opinion, a school of thought is that they cannot take and metabolize glucose into energy, so they depend upon astrocytes for that. So what astrocytes do is they take in glucose, they convert it into lactate, transfer the lactate into neuron. But this lactate is like a kind of a dicey thing also, because too much of lactate accumulating in the neuron is also bad. So lactate needs to be converted into energy very fast by neuron, and in aged neurons or in a neurodegenerative disease, lactate accumulation happens, pH changes, oxidative stress increases in the neuron. So lactate accumulation is not a good thing for neuron. However, neuron depends on lactate for normal energy metabolism. That's one thing to remember. And then the other thing is, we were interested to look into the promoters of GnRH gene, because we were looking into non-hormonal regulators of neuron. And typically the question we put forward is, I told you that GnRH all across the life is like a sine curve. So why the reason of no GnRH during aging? Now, is there a non-hormonal regulation? That's what we wanted to put the dots in. And we were looking into the transcription factors which are actively known to bind to the promoter of the GnRH, and we found out some of the transcription factors which had proper binding site to the promoter of the GnRH. And then we fished out one single cell RNA-seq from the aged astrocyte, and we did some reanalysis of that study to know how this crosstalk between astrocyte and neuron which secretes GnRH happens during old age, that is responsible for lowering GnRH at old age. And we found out from the pipeline that we did that there is a lactate that goes from the astrocyte to the neuron. This lactate continues to become accumulated in an old aged neuron, which causes increase in a pro-inflammatory cytokine, TNF-alpha signaling, and that further upregulates these NF-kappa beta and other genes which are pro-inflammatory again. And then finally upregulates all the repressors which are important for repressing GnRH gene expression, and inhibits the activators. So this was something which was published in this particular crosstalk that we talked about, that how independent to hormone the astrocyte can crosstalk with a neuron and then downregulate GnRH synthesis. The same thing is seen in a neurodegenerative disease brain. So in Alzheimer's and Parkinson's, it's well known that there is an associated lowering of fertility, which might be the reason because there is an increase in lactate from the astrocyte to the neuron, and this might be the reason behind it. Then we were further interested to look into the other novel regulators. So we were literally posing a question: if there is a aged brain-like condition, can we reciprocate fertility in that stage? So we were trying to increase fertility of those people who had a submerged GnRH level at a young age because of high amount of lactate or high amount of TNF-alpha in the brain. So in looking into the literature, we came across this novel class of biogenic amines known as polyamines. Now these polyamines, the word came from the fact that they have more than one amino groups. There are basically 4 polyamines, out of which the 3 that we were interested to look into, putrescine, spermine and spermidine. They are all de novo synthesized in the body by biochemical pathways, where they are all intermediates from the amino acid metabolism, methionine. Please.
So one small announcement, and many participants can go to their poster for revision.
So quickly I can wrap it up because I don't think there will be any remaining. So the thing is, anyway,
Good listener, don't worry.
I have already made you listen a number of times. So that's what I like to change the talks.
See,
But
I'm
anyway,
still interested.
okay, thank you. As fresh as your shirt. Yeah. So the thing is, arginine, methionine, and proline, they are metabolically acted on by several enzymes, which gets converted into the nucleic spermidine and spermine. This is not much research. What is more known is this same arginine goes into ornithine, ornithine goes into urea cycle, that all of us know from our biochemistry knowledge. And the other important part, a number of, I think one of this earlier speaker had talked about nitric oxide and NOS, ENOS, that is also related to arginine metabolism. So another pathway which is also related to the similar this amino acids is this polyamine pathway, where not much of research has been done. And so polyamines we want to regulate GnRH. Where from we discovered this hypothesis? Basically, information known till now was polyamine regulates gonads, gametogenesis as well as steroidogenesis. Polyamine regulates pituitary. So we imposed the question, why not polyamine regulate GnRH? Because it's on the top, it might regulate directly. So we started venturing into it, and the first model we used was very simple because nothing was known in this field. However, polyamine regulating other brain function is well documented. Hypothalamus was not touched upon, maybe because of us to venture in. So we use the ester cycle model, which is a reproductive cycle, normal natural reproductive cycle of a rodent, and across the different phases of the rodent reproductive cycle, we tried to map the expression of the polyamine associated factors and correlate it with GnRH. It's all done in the hypothalamus. So we obtained some correlation that I'm showing you later. Another model that we took was a simple aging model, because in aging model, just like the ester cycle model, only during a particular phase your reproduction is there, otherwise there is a low GnRH. And in that same tissues, what is the expression variation in the polyamine associated factors was something which we were looking into. And we obtained a more or less a similar kind of correlation, that in both the models there was a positive correlation between polyamine and the GnRH thing, both in ester cycle as well as in aging. What did this mean? This means that there might be that polyamine can regulate GnRH, because whenever polyamine is going up, GnRH is going up, and vice versa. So we then went into both cell line model as well as the animal model, where we treated. This is a cell line model, so we have the immortalized neurons which are grown in the cell, and these are specific hypothalamic neurons which, when treated with any positive regulators, they increase GnRH expression. It's well known. So whenever this is the polyamine that we use, the different doses of polyamine, and then this is one of the doses where this could increase the green spots. Green spots is GnRH expression. Both in RNA as well as protein, we saw the same thing. We took it into the animal, we injected polyamine in a normal female cyclic, adult cyclic female, and we saw that in comparison to the control, there was an increased expression of GnRH when polyamine was treated. We used inhibitor of polyamine, there was a decreased GnRH expression. So there was a positive correlation, there was a positive action on the neurons to increase GnRH. Then we went back to address our original question. In presence of an aged brain, can polyamine increase GnRH? That was the question that I started with. Then we created aged brain-like condition using two regulators of aging brain. One is lactate, one is TNF-alpha. So we treated the neurons with lactate and TNF-alpha. And this is a control neuron's GnRH expression. In presence of lactate as well as TNF-alpha, there is a decrease in GnRH. Because I told that GnRH, whenever there is high lactate, high TNF-alpha, GnRH expression goes down. In the same presence, when we added polyamine, this was the interesting finding of it, we could rescue GnRH. So adult brain में तो GnRH को बढ़ाती है. Even in aged brain-like condition, polyamine could increase your GnRH expression. That was something very interesting. Both in protein level as well as in the RNA gene expression, we found the same thing. अब ये चीज़ जब भी मैं present करने जाता था कभी seminars में, तो people used to pull my legs. आप ओल्ड एज को जवान बनाना चाहते हो, ऐसे ही भारत में प्रॉब्लम है इतना फर्टिलिटी को लेके. So this is something you are like literally going against what the government might be thinking. But I wanted to tell that polyamine has that power to regenerate fertility. तो तब तो मजाक मत बना के छोड़ देते थे. I couldn't help because there was no publication available. And to my rescue, then this publication came which was in Nature Aging. We all know how an important journal this is, and they told the same thing. Polyamine could rejuvenate, fertility in a senescent mice. If you break down the thing, exactly what we were trying to say, same thing they were telling. We immediately congratulated the author because Ziong, I had met him in one of the conferences abroad, and we know each other. So I told that it's a very nice thing. Not only that you had told something very interesting, you had rescued our lab work because there was nobody to support us. All were telling them no, ye ho nahi sakta hai. But this came up as a very interesting study. We had another thing in mind which we did it, and the other work what Ziong was doing, we couldn't do it in mice because at that time I initially was in Goa, BITS Pilani. I recently joined my alma mater in January. So there we didn't have the permission to work on the old age mice. So I couldn't do an in vivo work. Whatever GnRH I showed was in vitro. And Ziong did it, of course, in a in vivo condition. So his work had a greater impact, no doubt about it. But it was in line with what we were telling before we published this in Nature Age. And then the other age group that we tackled was the young age group. It was just 1 week old female mice. We treated it again with polyamine. What we saw was that, I'll just go to, so I might have missed the data. I might have deleted, I was reducing the number of slides. So what we got when we treated this 1 week aged mice with a 5-day regime of polyamine treatment, their ovary size increased and there was early onset of puberty on 14th day of its life cycle, whereas puberty comes around 25 days. Puberty comes around 42 days in case of mice, and our case we got it within 14 days. It was very, very early onset of puberty, ovary size increase, early markers of folliculogenesis, markers of your early onset of puberty in the hypothalamus, and markers of folliculogenesis were highly increased in the ovary. Because when my student first showed, I was literally doubting the result, and we repeated it at least 2 times with 2 different sets of pups. We got the similar kind of data that no, polyamine has that rejuvenating power. And then of course that 2023 Nature thing when it came, then we got little bit calm that okay, what we were doing is not in a wrong direction. So this also got recently accepted. So then the general question comes again that you are talking about polyamine having this impact on GnRH, but what is the mechanism? Can you talk more about it? So what we did was that we wanted to look into the transcriptome of the neurons after polyamine treatment in the GT17 cells. So we did a culture of the GT17 neurons in presence and absence of polyamine, and then we had sent the cells for RNA-seq. We had sent the total RNA, and then what we got, we plotted it in the volcano plot, and the pathways that we got. One of the hits, of course, we got from the RNA-seq was that polyamine upregulates GnRH. So again, it substantiated what we were telling, but we were interested in looking into the mechanism, the pathways. One of the pathways which was significantly hit due to polyamine treatment was the circadian rhythm. Now that was something which we also got in this cake pathway interactome that we found out that there was of course GnRH secretion that was hit after polyamine treatment, and also the circadian rhythm, the circadian enrichment. So then we went back into literature to find out what is the link between circadian rhythm, polyamine and GnRH. Now there is well known that GnRH in the female body as well as the male body is released in a circadian path way, that it goes up and down, up and down. It's not always up, it's not always down in a normal adult male or a female. It is also well known that polyamine also goes up and down in the brain. It's up and down. It's not always up, it's not always down. And then a recent paper to our rescue came that it was published in 2018. So what we were doing was polyamine treatment किया, GnRH बढ़ गया. बीच में क्या हो रहा है? That was the question we asked, and there were references. Circadian rhythm controls GnRH, polyamines control circadian rhythm. ये सारे available था literature. We just stitched this one after the other. That after giving polyamine through the circadian enrichment, there is an upregulation of GnRH production. So we got to validate this RNA-seq data. So what we did a circadian study we performed on the GT17 cells. 6 hourly we harvested the RNA, and then we checked for some of the circadian gene markers as well as correlated it with the GNRH. So GNRH graph, I missed it out here again. Oh no, it's here. So if you see the markers, then you will find out that the more or less the pattern of the expression of the circadian genes, they map with the pattern of expression of GNRH, which is like trying to substantiate the fact that after polyamine treatment there is enrichment of the circadian rhythm in the GT17 cells via the this upregulates or activates the GNRH secretion. Fine. And then we validated it in a mice model also, because we immediately made some collaboration. But here one work that is yet to be done, which we are planning, is to we are purchasing a plasmid which might silence one of these the major circadian clock gene. Particularly we are targeting the BMAL and the clock, these two genes, which are very important circadian genes. And then we are going to put this polyamine treatment, repeat the 6-hourly regime, and then finally prove that it's happening through this. Because this is just evidence, but in order to prove it more further downstream, we got to show that yes, by silencing one of the important components of the circadian rhythm, then giving polyamine, it doesn't increase GNRH. Then it's a full proof. We further collaborated with a person who works in circadian rhythm model, and luckily they had chrono-disrupted mice. My friend Dr. Devkar from MS University, Gujarat, Baroda. So he gave me the circadian disrupted hypothalamus tissue, and how it was disrupted was this regime that they had followed. It got published by them in Facet journal. They did some circadian study. We utilized their same tissues, and we wanted to check that in a already disturbed circadian rhythmic mice model, what is the scenario of polyamine genes and what is the scenario of GNRH. So we saw that the rhythm of the GNRH as well as the polyamine gene completely got disrupted, again trying to indirectly prove that all three components should be very importantly associated for a proper regulation of GNRH by polyamine. Fine. So this is what from this last part we concluded that through the polyamine treatment there is a regulation of the circadian rhythm in the hypothalamic neurons, which further activates the GNRH expression. So that is all. This is a work which was entirely done in BITS Goa. Goa is a place to go and roam around, but it can also be a place to work. That's what we always say. And I'm continuing this work obviously in BHU Zoology department after I joined. And these were my collaborators. A new field that I had started also recently, I hope I'll be able to talk about this and people don't get bored listening to the same kind of work that I do, is genital tuberculosis, because Ruchi, we got a recent grant from ICMR. We are going to look into how much closely associated TB infection with reproductive disorders. And there is a high correlation between the two. That's what we are trying to decipher the link. And these are, as Amresh told, all the all my heroes, heroes, I take his word. So these are the ones who did some part of the work. Major part was done by and Subrato. He is taking this work further ahead, because none of the funding agencies nowadays give just fund for basic work. We are trying to look into, and we got very interesting data with polycystic ovarian syndrome and polyamine. So that is something which he is taking care of. And these are my other team members. Thank you.
Now the session is open for questions.
Yeah, please.
I had a simple question.
नहीं आपका generally मेरे लिए specific है. हाँ.
When you are talking about female infertility, right, and then polyamine helps in making the animal more fertile in terms of the variance. Infertility is a broader term. So is that this effect of polyamine is in a given scenario, or irrespective of the type? Irrespective of the type of infertility which the females suffer, this will work.
Yeah, very good. So obviously infertility is a very big word, and what had been shown by Xiong's group was they had just taken senescent mice, and in senescent mice they showed that fertility is decreased because it's an aged mice, and in them they increase the number of pups. Functionally they showed that. So that's what we are also trying to say, that both cases, if you are either you are that work we have not done, but we are planning also that in neurodegenerative diseases, it's not a direct class of infertility in female, but in case in such kind of a disease, it might be. And one infertility type what we had done, I showed polycystic ovarian syndrome, which is a cause of to some extent infertility. There also, but very interestingly we showed was the PCOS induced mice, they did not mate. Whereas the polyamine treated PCOS induced mice, there was at least 50 to 55% mice which mated and they gave live birth. So that at least I can tell you.
Also in this circadian rhythm publication what we referred to.
It's not published as of now. Yeah.
Not the ones which you referred that publication.
Where
polyamine. So if you understand in a layman term, once the aging sets in, right? Like even if you take an animal model, is the polyamine able to revert it? A. And B is when if you start treating the animal at different ages, like at the reproductive age, then it becomes a little bit aged, and then sometimes when it has passed that reproductive age. What is the effect of polyamine?
So the first question, it's documented the circadian rhythmicity is hampered or altered in aged brain. So if this is altered,
and this polyamine is able to revert it once it has set in.
Yeah, that is what I did not do it, but that's again something we are planning to check, that whether in the brain the circadian rhythmicity they are able to bring it back by polyamine treatment. Because with Devkar, the work that I showed, we got autopsied tissue samples. There was nothing, no place to inject. Otherwise, I had a plan that circadian disrupted mice model.
That model is like a temporary.
It's a temporary model. Yes, of course.
The aged one is
Yeah.
permanent.
As I told you, the aging data I showed was in neurons. So if we had the mice, aged mice, and injected polyamine and then checked the rhythm is coming back or not. But as per the data of Xiong and as per what we obtained, high chances are there is a a restoration of circadian rhythmicity. High chances.
Same line of question.
Please.
Like I was interested to know, like you have given polyamine. You have given polyamine. Okay, and the ovary size has increased. So is there polyamines have some kind of impact on the metabolism also? How it has increased the size of ovary within few days? Like you said,
the size might be, I might be wrong, you are correct, because and what we showed was folliculogenesis increased, and in the section the the number of follicles, they were highly increased. Whereas in a 1 week old mice plus 5 days is hardly how many days? 12 days. 12 to 13 days. You don't get any follicles there. They are all in their gonocytes and they are just multiplied by mitosis. And all are stuck in meiosis if you talk about the oocyte. So there is no such increase shown, follicles shown at all. But here we literally showed follicle. That might be the reason that the size increased, because follicles are forming, there is antrum, everything was there.
Is there any impact on the metabolism as well?
Metabolism,
like ultimately what is happening, either your body is building up few things to make the system ready for reproduction?
Maybe because hormonally they are increasing. We got an increase in GnRH, I didn't show that. After young 13 to 14 days mice, there was an increase in GnRH. There was an increase in some hormones other than GnRH, LH or FSH, one of them increased. FSH increased, which was not supposed to be. It was all of quiescent stage.
Like what my understanding says, so GnRH pulse comes in your pre-pubertal age also, right? After that subside, and once you reach the puberty, again it started.
I don't think pre-pubertal it comes. It might be very, very young. I don't remember whether there is any. But
I am not expert in that.
Puberty में आता है. Puberty में आता है. It was an early arousal of it. So, and one thing that
let me reach to the question. My question was whether this is a transient peak you have seen, or it's like once GnRH has started, it is leading towards the end of reproduction or successful reproduction.
There is no GnRH peak at all at the time when we checked.
Okay.
That made them awake. Early awake of them. It's like an alarm which is not to ring at that time.
Thank you.
So I have one question.
Yeah, please.
How do you suggest that the model that has been created for clonal disruption has been validated? Because as far as I know, melatonin is the main molecule which marks the circadian timing to the different organs in the periphery. And in case of black mice, the gene involved in melatonin is truncated.
So I don't know much about the validity of the model because it was done by Rajneesh and they had published it in Facet, so I might have to just forward you the paper that whether all these markers have been kept in mind. Because they had told that word that it's a chrono-disrupted mice. I just used their model issues.
So did they check the level of melatonin?
I don't remember. I have to just refer to that paper. Because I have not checked. I have not checked. I was just given the hypothalamic tissues. I wanted to see the chrono disruption has happened and then what is the effect.
Thank you, sir, for your enlightening talk. Now I would like to invite Dr. Sameer to frustrate Dr. Banerjee, sir. Okay, sir. It's my pleasure to introduce Dr. Gyan Prakash Modi, sir, Associate Professor at the Department of Pharmaceutical Engineering and Technology, IIT BHU Varanasi.
Already we are running late, no need to long introduction. I'll come to topic directly. Novel NIRF diagnostic agents for Alzheimer's disease. I'm not sure many of you are aware or not, this is a hardcore chemistry. So I may be boring, already running late, so top of that boring, double, Anyway, so we know about the Alzheimer's disease, the multifactorial and so many factors are associated. Many of you might be aware about those factors. One of the very important factors which comes into the proteins. But before that, I'd like to highlight one important point about the disease burden. There is an interesting fact that disease burden is going to increase in the developing country more compared to the developed country because this is a major issue in the developed country. But this can be a more problem in the developing countries. So this is really a because of change in our societal structure. Regarding the pathophysiology, two importantly is the acetylcholinesterase and glucosylcholinesterase along with the blood beta protein and tau protein. They play a key role. And as you know very well, this is a healthy brain, this is in case of the advanced AD, and these are the various factors. There are a lot of other factors as well. So coming to the treatments, when we look at the marketed drugs, some of the drugs which are the cholinesterase inhibitors like donepezil, glutamine, rivastigmine, and the memantine energy receptor and new monoclonal antibodies are also coming. But these drugs are effective only for short duration. Obviously, the disease is complicated. So what is the major challenge in Alzheimer's is also challenge in many diseases, the diagnosis. That is the major challenge because diagnosis happens when already 80% neurons are gone or so that time any treatment which we give probably not very effective. One of the major challenge. So if we look into for the early diagnosis approaches, there are few ways by which the disease can be diagnosed at early stage but still nothing is in the clinic. One of the very important and commonly known PET probe is Pittsburgh probe PID, which is one of the well-known probe which being used in clinic as well. Along with this, the new NIRF, near infrared fluorescent molecules, these are also coming up. Still, no one of these molecules they are in the clinic because there are some sorts coming with these molecules as well. But people are trying a lot. So we are already working on therapeutic agents development inspired from the natural product funded by the CRB. So while working on that project, we realized that diagnosis is a major challenge. So obviously we thought to start work on development of the diagnostic agents. So that's a project we got from ICMR to develop the novel diagnostic agents, development of the novel NIRF probes. We already had the molecules in our lab from the therapeutic So we thought to actually look into literature, already paper published in Journal review article. So we thought to cyclize these molecules and try to develop in such a way that we can detect the disease possibly in the animal brain, preferably in the eye, because there are evidences indicating that the protein aggregates start appearing way earlier in the eye of the retina of the patients. There are a lot of evidences behind that. So that's why Raman spectroscopy has been also widely used for detection of these aggregates in eye. So based on this diagnostic approach, we worked quite a bit, and we already filed a patent and published it in sensor and actuators, where we have shown the diagnostic probes work very well. But again, as I said, there are limitations with these probes still before they enter into the detailed clinical studies. So then we thought let's enter into the theranostic agents. Can we have the diagnostic as well as therapeutic together? So as I said that we are working on this natural product, we came up with this lead molecule. We thought to follow a hybridization approach. So we took up this fragment, but rationally and systematically modified in such a way that we came up with the new molecules. Again, I'm not talking about detail of the chemistry, because that will be too long. So this is the approach how we synthesize these molecules. These are just representative molecules. And these molecules we developed and characterized through various spectral techniques, and then we, obviously these are the NIRF probes, so we went for their excitation and emission measurement, and we realized that they are actually having their emission, some of are beyond 700, which is still not the NIR, but it is close to NIR. Then we made, as we're working in the Alzheimer's area, so we know how to make the different aggregates of the cellular beta proteins, 14, 42, or also of other proteins like alpha-synuclein tau protein. So from there we determine the selectivity of these molecules against specifically A beta 40 and 42, and we have done a lot of other experiments. The selectivity determination was done with the tau protein as well as the alpha-synuclein protein, their various forms, and the safety was studied in the cell line model. And then we also try to find out whether they have the property to disaggregate the A beta aggregates, but they do not have, except for A beta 40. 42 they are not able to break the aggregates. Then this is the APBPS1, the wild type mice, and this is the APBPS1 double transgenic mice of 13 months and 6 months. There's the reason behind selection of these age groups that we can discuss later if required. And we wanted to see whether these compounds has the ability to detect these aggregates or not. So that was actually done, and we also used the OC, which is also well-known dye, to detect these aggregates available in the literature. And then we also measure how deep they can go into the mice brain. Then again we did the study and compared with other well-known established dye like hydrofluoric S or like for the granite, which was developed by the man. And then we compared with in the various experiments to show whether they are able to detect these aggregates or not. So this is a component dye, hydrofluoric S. And these are again with respect to the antibody with 542, and these antibodies are specific against different type of the aggregates. Not again going into detail because time limitation. And again now the next study we went with the area autopsy samples, 92 years age female, not disclosing the more detail. And but we observed that the granite 2, which is already established in the literature, and hydrofluoric T, we compare to that, I43 has some superimposition, again not exactly, but yes, there is some superimposition. This is with respect to the, again in autopsy samples, but with respect to the antibodies, again we can see there is an overlap with the detection of this probe. Then this was also used in the mice model in APBPS1 mice, APBPS1, which is double transgenic live animal imaging to see whether it can detect, enter into the brain, detect A beta aggregates or not. And it has that ability to detect these aggregates. Then again, detection of these aggregates in the different tissue samples. Detection of these aggregates, selectivity is a major challenge. This compound has selectivity, but still is not 100% selective. So this is with respect to the alpha-synuclein staining in the 3D autopsy samples. And then we went for the enzyme inhibition studies. Compound has an ability to inhibit the enzymes as well, data are shown over here. We did the brain permeability study and then the ability to cross, provide the symptomatic relief in scopolamine-induced amnesia model, and that was in two different animal models. I'm just putting only one model here. Then we did also the detection of these aggregates in the Drosophila model, and what we can see here, this is an ability to detect these aggregates in the Drosophila. Again, experiment was done in two different ways. I'm not discussing detail over here. So we are also working on therapeutic agents development inspired from the natural product. These are few data which are being summarized over here. I don't want to get you more bored now already, 6 o'clock. So please accept my apology. These are the lab members who has been extensively working, and these are my collaborators, Dr. Sarut, obviously I should name them, they are very helpful. Dr. S. V. Krishna, Sarika Gupta, Sairam Krishnamurthy, Bonga Red, and Prabha Garg. These are my mentors, and I am very much thankful to SCRB, ICMR, and thank you very much to the organizers. Thanks for listening. I think I finished in time. You can ask that. Mic is not required. But
one particular question I answered a little less. So I'm just interested, can you make some kind of molecule which can let us sustain release, I mean the independent release of a drug, so to cure a disease or for a particular parameter. So is it possible with your system?
it depends upon me. You want to make a new molecule?
No, it's a known one. Known drug is.
So then we can go for the formulation. We are already developing the formulation for our systems. Okay, any questions? Students, right? Thank you very much for questions.
So it's just not a question, it's a discussion. So what do you think, like, how soon we can see a molecule accurate diagnostic possibility? I guess. I remember challenge is not as I attended a symposium back in 2019 when there was a very famous AD discussion politically. So we said that in 2025 we will see some already there. So complicated. But nice one. Yeah, because we will dive into diversion.
We can discuss it. Yeah, we can blame.
Thank you very much, sir, for your wonderful presentation. Now I would like to invite Dr. Ravi sir to felicitate Dr. Gyan Prakash Modi sir. Now I would like to invite Dr. Vinod Tiwari sir, convener of the event SNCI Neuroscience Symposium, to kindly share concluding remarks of today's session.
So thank you so much, Nivedita. So it was a very busy day. We had a very tough task, of completing, all the talks, including poster session, one time. But fortunately, we were able to do that timely with some little delay. So I would like to conclude today's session with sincere thanks to all the esteemed speakers, our chief guest, Dr. Arvind Rao, Dr. Ashita Banerjee, Dr. Anurag Vashne, Professor Supratap Singh, Professor Tripathi, Professor Ashutosh Dubey, Dr. Amresh Kumar, Dr. Gaurav Kumar Pandey, Dr. Gyan Prakash Modi, all the faculty members, especially the enthusiastic participants who have joined us from some parts of the country. And along with that, I would also like to convey my thanks to Dr. Rajneesh. With this, we come to an end of today's session of SNCI Symposium, and we will continue the next part of the symposium tomorrow morning, sharp at 9 o'clock. Okay, at the same place as we are here today, right? So please try to come on time. Don't come at 9. Try to come at least 10 to 15 minutes before 9. Okay. And tomorrow we'll begin the day with a keynote talk by Professor Alok Kumar, who is a professor in the Department of Molecular Medicine and Biotechnology at Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow. He'll be sharing his insights on cutting-edge developments in molecular neuroscience, and we are looking forward to your presence for this impactful session. And when you are in Varanasi, try to visit the places including Vishnu Temple. We have one temple, people who are coming from outside. We have one Vishnu Temple inside the campus as well. Right now, there is a situation of kind of flooding, so avoid, because there is no boating right now. I think Ganga is also banned right now. So do not go to the, in this right now, whatever the comment. But yeah, if you have some other plans, visit the temple, you can visit Sarnath or some other place. Thank you so much everyone.